We got video? Okay, perfect. So good morning and welcome to the clinical session of FlowTex this year. To everyone online and in the room here, we're just always really excited to hear the talks, but really just to have you in the room. And that's what I wanted to start saying today. We've been doing this for about 18 years now. The FlowTex crowd has been showing up and having these meetings focused on cytometry. And this year, I just really wanted us to focus on each other, because the only way that we have made it is together. And I know in the times that everybody's facing now, things seem uncertain, but I just wanted to give you guys a moment, and we'll take a moment of silence to just really appreciate that we are cogs in the wheel, and that as we find that inner calling and that inner voice, we can continue to move forward no matter what is happening chaotically around us. And so just take a minute to do that, and then I'll introduce our first speaker. All right, so our first speaker, he's here from an international talk, so he's staying up in the evening to share this information with us. He actually was a speaker at ICCS this year. Julian is a senior R&D scientist with a focus on assay development for therapeutic modalities. His work spans various applications, including immunology, dermatology, allergy, oncology, and all intersecting on compound interactions in whole blood, immune cell components, and complement proteins. Julian holds a PhD in T-cell immunology from the University of Amsterdam, where he conducted research on T-cell biology and effector functions. During his doctoral studies, and even after, he developed and implemented innovative flow cytometry assays, especially in the clinical realm, and that's what he's here to talk to us about, FlowFish. And this allows for the simultaneous measurement of cytokine, mRNA, and protein in T cells. So Julian, with that, I will turn it over to you.
Awesome. Thank you, Marcia. I hope everyone can hear me okay. I'd like to start by thanking the organizing committee, and in particular Marcia, for allowing me the opportunity to talk to you today. And as Marcia already introduced, I would like to talk about one of my longstanding research interests, which is FlowFish, and how to use it beyond its more typically used application, studying telomeropathies, and indeed employ it as a research diagnostic and clinical tool. So with that said, I have no conflicts of interest to report. And today I'd like to talk about what is actually FlowFish and what is it generally used for. Then I'd like to make use of the data that we put out there on our FlowFish study, how we set it up, starting with FlowFish design, and then how to prove that you're actually measuring what you think you're measuring. And then I'd like to dive into additional novel applications of FlowFish and what you can think of using it for. So to start off with, what is FlowFish, as the name implies, and my presence at this conference? It's a flow cytometric technique. It allows for the high throughput measurement of single-cell specific RNA or DNA sequences, and this can be anything that's made up out of nucleotides. So you can think of telomers or other repeat-like structures, mRNA species, such as mRNA for the cytokine interferon gamma, but also microbial genomic material, such as HAV1. And FlowFish can be used at the same time to assess RNA or DNA, but also protein via classical flow cytometric antibody staining. So you detect your protein of interest with fluorescently labeled antibodies, and you can detect your DNA or mRNA species of interest with fluorescently labeled probes, or probe labeled with a tag that you can then detect via either click chemistry or antibodies as well. So currently there are a couple of commercial kits available, but also homemade protocols are out there to use for you. A typical FlowFish workflow looks as follows. First, of course, you isolate your cell type of interest. This can be primary immune cells or neurons or paraffin-embedded tissue. If you're looking at an inducible gene, you first activate your cells. If not, of course, you can forgo this step. Then you proceed to do the surface stain so that you can identify your cells of interest in a mix of cells. And for this, you can use classical antibody staining, as I said. Then you fix the cells, and this is a critical point because at this point, the nucleic material becomes accessible to DNases and RNases. In our case, we were interested in mRNA, so all compounds that we used after this, we pre-treated with RNase inhibitors to make sure that we didn't destroy our target mRNAs. So after preparing the probe sets and all other reagents, you can then perform your intracellular stain and your fish stain, which typically occurs overnight, which is a nice break moment. And then the next day you come back to the lab, you wash your cells a couple of times profusely, and then you measure by flow cytometry. So for all the plots that I'll be showing, mRNA will be on the y-axis and protein on the x-axis. And you can then gauge on populations of interest, of course. So originally, FlowFish was set up as a technique to measure repeat sequences, and specifically telomeres. And nowadays, it's actually most commonly used still to investigate telomeropathies. And in general, how this works is you take your patient sample and you spike in bovine thymocytes, for which the telomere length has been quantified via traditional methods. And then you add either the probe or not, and then you can look by fluorescence what the probe length is by making a ratio between the bovine thymocyte signal and the signal of your cell type of interest. And by making use of sequential gating and fluorescently labeled antibodies against extracellular markers, you can then zoom in on your populations of interest. So in this particular study, the authors used a female reference sample and a male normal donor to investigate the telomere length in a patient with a suspected telomere disease. And if we then look at how the control cells look, with in green the unstained sample and in red the stained sample, you see that there is a nice big difference between stained and unstained, which you can then use to quantify the telomere length in the human cells. So if you gauge on populations of interest, such as granulocytes, you see that there is a difference in staining between stained and unstained in the female reference sample and also in the male normal donor, but that this window is largely reduced in the patient with a suspected telomere disease. Similarly, if you gauge on B cells, what you see here is a normal distribution of telomere length in B cells in the reference sample and in the normal donor. And you see that these cells with long telomeres are completely absent in this telomere disease suspected patient. And indeed, it was later shown that this patient had a telomerase deficiency. However, FlowFish can be used for a myriad of other things, basically any nucleotide bearing sequence of interest that you would want to investigate. But how do you go about setting up a new assay? So for that, I would like to make use of some published data that we put out there, which centers around T-cell immunology. So as hopefully most of you know, T-cells play a pivotal role in antiviral and antitumoral immunity, where naive T-cells are activated by the cognate interaction between the T-cell receptor and an MHC molecule bearing a matching antigen by antigen-presenting cells. When there is a match, the T cell becomes activated, it will expand and it will move out into the periphery, where upon encountering an infected or a tumor cell, it will start to secrete large amounts of infector molecules in hopes of killing the infected or tumor cell. These canonical T-cell effector molecules like IL-2, interferon gamma, and TNF-alpha can be easily measured via protein-mediated detection. This can be either through ELISA or it can also be via flow cytometry, by intracellular staining. However, harder to stain for molecules like IL-4, IL-5, IL-13, but also CCL3 and 4 can be more rapidly picked up with fish-based methods because the antibody tools for those molecules are not as developed as the one for the canonical T-cell effector molecules, but also because their production is often at a much lower magnitude. However, in general, T-cell effector function is governed by post-transcriptional mechanisms in both health and disease, resulting in a discrepancy between the amount of mRNA and protein that you can measure. Furthermore, this discrepancy can even result in copious amounts of mRNA being present without protein being formed. This allows memory T cells to respond rapidly upon infection, but this is also a characteristic of dysfunctional tumor infiltrating lymphocytes. And probing these mechanisms is exactly why we set up this method, because we wanted to further dive into these mechanisms in hopes of finding out more about T cell biology and looking for potential therapeutic approaches. So to do so, we set up this flowfish assay. And the first thing that you do, excuse me, is design your probe set. And for that, you can use perfectly good commercially available tools, such as the Stellaris Custom RNA Fish Probe Design tool, where you put in your sequence and then it spits out a probe set of interest. Optionally, after that, you can use BLAST to control for species specificity, and also to investigate whether any off-target effects are expected. You can look at the expression of secondary target genes. For instance, if you're designing a probe set for IL-2, but one of the probes that is designed partially overlaps with a gene that is only expressed in osteoclasts, it might not be that much of an issue to include such a probe in your probe set. All right, once you've got your probe set designed and produced, what next? Well, you put it to the test. So we designed a probe set for human cells and mouse cells for interferon gamma. And if you activate human cells, but also mouse cells with PMA ionomycin and then stain for interferon gamma protein and then counter stain with the interferon gamma mRNA probe set, you see cells popping up that produce both, but also cells that are only mRNA positive or protein positive. Because we put out more data on the development of the mouse probe set, I'm going to show you how we validated this mouse probe set, but we did exactly the same for our human sets. So the first thing that you do, of course, like you do with any new reagent, is to figure out what is the right concentration to use. So we took a huge batch of T cells, we stimulated them, and then we added increasing amounts of the probe set. As you can see here. And what you can see is that the signal for interferon gamma mRNA goes up if you add increasing amounts of the probe set, which is good. But at some point, you also start to squeeze the signal between positive and negative, or lowly positive and positive. And therefore, we ended up selecting the 50 nanomolar concentration for use, which is also coincidentally what the manufacturer recommended. So that's great that that fits. The next thing that we did is to look at specificity. So to do that, we made use of other probe sets that we designed. And in this case, for looking at the specificity of the interferon gamma mouse probe set, we made use of a human probe set targeting the FOX2B gene, which for us at the time was a gene that is not expressed in immune cells, but in a different target cell type. But it was labeled with the same fluorophore. So if we then took mouse T cells and we activated them with PMA ionomycin, we stained for interferon gamma protein and then counterstained either with the FOX2B primer set or the mouse interferon gamma primer set, what we see is that we get a nice signal for the interferon gamma primer set consisting of mRNA single, protein single, and double positives. But for the FOX2B primer set, we do not get any signal in the mRNA channel, indicating that our probe set is at least specific, or it seems to be specific. And the nice thing about using this type of approach is that you can actually use this FOX2B primer set as an isotype control as you would for a normal antibody staining. So next, what we did is something that is called a cold probe assay, where you take your probe set and you add equal amounts of labeled and unlabeled probes and then skew the ratio towards unlabeled. Indeed, adding more unlabeled probes. And if everything works as you think it should, you should slowly see the signal titrating out. And that's exactly what we see. When we add more probes here in a 5-to-1 ratio, here in a 100-to-1 ratio, we see that the signal slowly goes down, indicating again that when we oversaturate with similar sequences that are unlabeled, we start to lose out on the signal, indicating specificity. So we didn't only do this for interferon gamma, we also did this for TNF alpha and IL-2. And I hope you can appreciate that the expression pattern of the cytokines and their respective mRNAs is completely different, which again highlights the importance of looking at both mRNA and protein at the same time. So now you've set up your probe set, but what can you use it for? Of course, typically you would first look for your application and then develop a probe set, but story-wise, this just fit a little better. So the first application that I'd like to talk about is rounding up the T-cell effector function story. So a bit of our own data, where if we take human cells and we stimulate them, and we then select cells that are mRNA high or mRNA positive, and cells that are mRNA low or mRNA negative, and then look at the amount of interferon gamma protein that they can produce, what we observed is that mRNA high cells also produce larger amounts of interferon gamma protein, and mRNA low cells produce fewer amounts of interferon gamma protein. Conceptually, this all makes sense. But interestingly, if you then counterstain not for interferon gamma, but for TNF alpha or IL-2, we see the same phenomenon, indicating that these mRNA high cells are better equipped to produce cytokines in general. If we then flip it around and then look at TNF alpha, we see a similar pattern. Also here, TNF alpha mRNA high cells are more capable of producing interferon gamma and IL-2 protein compared to the mRNA low cells. And this is important because for vaccine approaches, but also TIL therapy approaches, you would want to have effector T cells equipped with the capacity to produce as much cytokines as possible, because it was shown that these cells that produce more cytokines, so polyfunctional cells, are actually beneficial for the patient. So by assessing both mRNA and protein at the same time, you can probe the regulatory mechanisms that govern this production in a high throughput fashion, and you can use it to design therapeutic approaches. So that is also what we used the mouse probe set for. But first, there is a bridge that I forgot about, because in the therapy landscape for cancer, there's, of course, CAR T cells, but also tumor-infiltrating lymphocyte therapy. And currently, the golden standard to release a batch of produced TILs or CAR T cells is an interferon gamma ELISA. Basically, this tells you about whether there are cells that can produce interferon gamma or not. However, to assess therapeutic efficacy or predict response or predict longevity of responses, it would be much more beneficial to have a little bit more insight in how your product looks. You would do this by flow cytometry, and why not assess everything in one go? Indeed, we would suggest to use something like cytokine flowfish to look at not only the amount of cytokine producing cells, but also whether they're polyfunctional, because that is beneficial for patient treatment, to look at how the post-transcriptional mechanisms are after reprogramming these TILs. But you can also think of other things like the expression of inhibitory receptors or homing receptors to get a clue about whether these cells can actually end up in the tumor and perform their function. So we also did use Flowfish to look at TIL therapy. And for that, we made use of our mouse probe set, where we took wild-type mice or mice in which the interferon gamma post-transcriptional regulation that I just talked about is decoupled. And while wild-type mice, if you co-culture them with a tumor cell, they can produce copious amounts of interferon gamma in a short period. They lose this capacity over time. While our del T cells, so the ones that have a deregulated interferon gamma production, are able to sustain this for a much longer period of time. If you then take tumor-bearing mice and you treat them with either wild-type TILs or our del TILs, we saw the following. While wild-type TILs are able to delay tumor outgrowth a little bit, our del T cells were significantly better therapeutic approaches and significantly delayed tumor outgrowth. So again, this is a way where you can use this technique to probe what is going on and design therapeutic approaches around it. So to swap gears a little bit, what else can you use Flowfish for? And I'd like to now talk a little bit about pathogen or microbe identification. So this is Flowfish can also be used to identify microbes, and these can be bacteria or fungi, and they can be in all different kinds of samples. Literature is full with research about that, and this is a summarizing table from a review, where, as you can see, indeed quite a lot of different bacterial species can be targeted with Flowfish, but the power of Flowfish comes into view when you identify more than one strain at the same time. Indeed, in diagnostic settings, the speed advantage of Flowfish, which typically takes 1 to 2 days compared to bacterial culturing, could be very beneficial. Also, not for all pathogens out there, culturing is available, and this can really take weeks. Furthermore, in contrast to other techniques, such as culturing or 16S ribosomal RNA sequencing, Flowfish provides information on the relative abundance. It tells you about which type of pathogens or microbes are in your sample of interest. Of course, on the other hand, the downside compared to 16S sequencing is that you can only investigate species that you can specifically target. However, given that many hospitals are equipped with a flow cytometry lab and 16S sequencing is a little bit more specialized, there is a window to incorporate this in a diagnostic setting. So to continue the story about how Flowfish is used in microbes, it's also been used a lot in virology. To look at what type of viruses are present, to characterize infected cells, and to look at their replicative status. And one major virus in which Flowfish has been used quite thoroughly is HIV-1. So the current method to assess HIV-1 levels are investigating most often patient serum for the presence of HIV-1 genetic material or HIV-1 virions. Sometimes they also investigate HIV-1 proteins or anti-HIV-1 antibodies. And another thing that it's being employed are assays that investigate replication-competent HIV-1 proviruses, which are sequential culturing experiments that are being performed. However, this is a highly specialized technique, and you can swap this out, for instance, with Flowfish, and it would give you, again, a different type of information compared to these traditional techniques. First of all, you know nothing about the cell type that is being infected. While HIV-1 mainly infects CD4 T cells, it's also capable of infecting monocytes or macrophages and dendritic cells. So by using Flowfish on the cellular fraction, you can figure out the amount and the type of infected cells. Furthermore, because this is a flow cytometric approach and it's multiplexable, you can investigate receptor or marker expression to figure out if these cells express different markers or particular markers that are targetable with a therapeutic approach. Patients who are on successful therapy, often antiretroviral therapy, they suppress the translation of the HIV-1 virus into protein. And flowfish can be used to look at both the latent reservoir, so how many cells are infected, but also how many cells are currently translating RNA or into protein. And lastly, as I alluded to already, you can also use it to look at therapy efficacy of either traditional therapy or novel adoptive cellular therapies. And one example of a flowfish application is this paper right here, where authors took samples from HIV-1 infected individuals and then probed which CD4 T cell subtype was preferentially infected or expressing HIV-1 RNA. And unsurprisingly, this is something that has been known for quite a while now. They were able to confirm that it's mostly central memory and effector memory T cells that harbor HIV-1 RNA. But this technique also allowed them to look a little bit deeper into the expression profile, to look for novel insights into HIV-1 biology. Because what they did next is some deeper phenotyping. And what they showed actually was that if you take either patients that are on antiretroviral therapy or patients that are even still viremic, and then you select for HIV-1 RNA positive cells, what they actually showed is that these CD4 T cells express low levels of CD20, which canonically is a B cell marker that is not expressed on healthy T cells. And indeed, CD20 negative T cells were not RNA positive for HIV-1. They even went a step further and used in vitro models where they treated patient samples with rituximab, an anti-CD20 therapeutic antibody that is used to treat B-cell malignancies and autoimmunity. And indeed, they were able to show a reduction in CD4 positive CD20 DIM T cells. And this type of phenotyping allows for novel insights into HIV-1 biology and identifying potential therapeutic targets. So another virus in which flowfish is being used quite often is the Epstein-Barr virus. Like HIV-1, EBV preferentially infects immune cells, which potentially can alter their function. It's something that not a lot is known about. And again, using flowfish, this can be studied because you can investigate which cells are EBV positive and then see if they have a different effector function compared to non-infected cells within the same sample. EBV also has a latent cycle that sometimes is disrupted, and you can look at this latency reversal also using flowfish. Similarly to HIV, you can also identify EBV-associated markers by using this technique, and you can also assess the killing of infected cells through therapeutical approaches by using flowfish. So an example of the type of data that comes out of this type of flowfish assay is shown here, where the authors took EBV-infected samples from patients who are EBV infected. They stained for EBV material, so EBER, and then subdivided in EBER-positive cells and EBER-negative cells, and then focused on the B cells, which are one of the target cells that are preferentially infected by EBV. And what they showed is that irrespective of CD19 high or low expression, the EBER-positive cells enriched for CD27 high, CD38 high, IgD low, indicating that the EBV virus preferentially infects or is harbored in a memory-like B cells. This is something that was new at the time, and again allowed for novel insights into EBV biology. EBV infection is also associated with the development of lymphoproliferative diseases. And depending on the cell type, you would need to have the cell type of the lymphoproliferative disease, you would need to treat patients with a different approach. So what is being done right now is that these cells are either muxed out by mux sorting or they're foxed out via fox sorting, and then their genomic material is obtained and assessed in which cell fraction the EBV DNA is present. However, if you're foxing anyway, why not immediately assess via flow cytometry? So that is what the authors of this paper also looked at. They looked at a host of different lymphoproliferative diseases, but I'm just going to highlight one, which are the hydroxyphosphatidiform-like lymphoproliferative diseases. So there are 3 patients here. They stained for the EBV probe set and then counterstained for a lot of different immunological markers. And what they showed is that EBV-positive cells did not express CD19, so they weren't B cells. They did express CD3, so they were T cells, but they were negative for canonical markers like CD4 or CD8, but rather expressed TCR delta 2, indicating that indeed these patients had hydroxyphosphatidiform-like LPD. And this allows the selection of proper therapeutic options, or can at least guide that. And then the last thing that I'd briefly like to talk about is something that Marcia put on my radar, actually. It's a paper that came out this year where FlowFish was used as a tool for sorting cells by flow cytometry. Because as you can imagine that, of course, for relatively abundant cells, it's quite easy to look at protein markers. If you want to do single-cell RNA sequencing of B cells, you just fish them out with CD19. However, if you're looking at rare populations, and with rare, I mean 1 in 200, 1 in 3,000, you would need to sort or send in a large amount of cells, which is, first of all, wasteful, and second of all, it clouds your the data that you generate. So the authors of this paper thought, why not use FlowFish to enhance these rare populations, which are often characterized already via RNA sequencing. They're often markers for which antibodies are not available. So why not employ FlowFish there? So that is what they did. They took complex cell form populations like total immune cells. They put in probes of interest and then used hairpin amplifiers, and I'll come back to why that's important later, with a fluorophore to get a signal that was sufficient for sorting. They sorted out the cells and then did single-cell RNA sequencing. As a proof of concept, they first started with B cells, and if you take B cells and just sort them on CD20 or CD19 and then put them up for sequencing, you get about 84% of reads that can be mapped to the genome and that are not mapped to the probe set that is used to fish out the cell types of interest. However, if you use FlowFish, the FlowFish tool, you get only 6% of specific reads, indicating that you get really a giant loss of information there. So the authors went through a lot of hoops to figure out what was going on. And actually, what was happening is that because of these hairpins, they were getting a lot of probe information back that was specific to their probe set and not to the genes that they were actually looking for. So they figured out that if you treat with DNAs after sorting to get rid of the probe set, they were already able to up the amount of specific reads. And then by swapping from classical sorting solutions containing bovine serum, which has RNases in there, to BSA, they were also able to up this to 90% of reads confidently mapping to the genome of the target cells of interest. And this allowed them to use fish-based enrichment of rare cell populations ranging from immune cells to neuronal cells obtained from both fresh, and this is of particular interest, also paraffin-embedded material, which is classically hard to get good single-cell RNA-seq reads from. So this just shows that you can use FlowFish for basically anything that you can imagine. And in view of time, I'll stop the presentation now. But FlowFish has been used also during the SARS-CoV-2 pandemic. It can also be used to assist in visualizing the impact of CRISPR-mediated genome editing. And much more. And that is all to you and your creativity. So I'd like to challenge you to go out there and employ FlowFish in your own workflow. To summarize, FlowFish allows for measuring specific mRNA or DNA sequences via flow cytometry. With proper experimentation, you can set FlowFish assays up against virtually any target of interest, and diverse fields have applied FlowFish, including studies surrounding T-cell effector function, microbe identification, viral biology, and novel therapeutic target identification. I'd like to thank my old supervisor, Monica Wolkers, and my two great collaborators, Benoit Nicolette and Fiona Salerno, with whom I've performed all the FlowFish studies. My new employer, Gaius, who was able to provide me with the time to prepare this presentation for you, and of course, all patients, blood donors, and mice included in this study. If you have any questions, I'm happy to take them.
Thank you, Julian. Very nice talk. First, I'm going to see if the audience has any questions. So if you've been with us any amount of time, you know I'm always strategic in putting who I do as speakers. And Julian, that very end point that you made, that paper was originally posted by Dylan from Single Cell Discoveries, who does a lot of RNA-seq for cell therapy development, so commercialization. I firmly believe, just like when I stood up here years ago, probably a decade ago, and told you spectral flow cytometry was the next thing, and we're about ready to hear a great talk on spectral flow cytometry. This is actually the next paradigm in cell analysis. And the reason is some of us are very familiar with LACE. They won the Cyto Innovation Tool a couple years ago. And what they're providing is the ability to look at signaling pathways. So Carl June was one of the first, and we have a talk from someone in his lab, was one of the first people to talk about how important signaling pathways were in CAR Ts, in the ability for patients to evade the CAR T therapy. So that ICOS signaling pathway that leads to TRAF2, right? So it's the metabolic state. So it's going to be important for us to do a 40 color. We've never really been able to look at sequencing pathways because we haven't been able to do that 40 color panel followed by 40 color phospho, followed by potentially live cell. That would be great, but even just fixed cell genomic material and looking at all of that data without having to make inferences across it. So I wanted to bring this up. We always think of FlowFish as, hey, this is like something that we used to do a long time ago. But what we're finding out is there's a lot of non-coding RNA that's important to the cellular environment, especially when we're going in and trying to treat CAR Ts. So Julian, you did a fantastic job setting it up, saying, hey, this is how we want to go about doing it. My question for you is, in your experience, when you've needed to fix cells to go in and do this, have you noticed that you mainly only get the nuclear RNA from that, or have you actually been able to get cytosolic RNA?
So that's an excellent question. The tricky bit about answering that is that I've worked mostly with T cells, which are famously known for being just a nucleus with a little bit of periplasm. We have used FlowFish also, well, not FlowFish, but we've used this probe set in imaging approaches as well. And there we could actually mostly detect the signal just outside of the nucleus. And we use the same fixation and permeabilization and everything for both the flow method and the imaging method. So I would say that indeed it's cytosolic mRNA.
Okay, that's interesting. And then, so you did mention that so one of the things, another, I guess, step forward is we've been talking about single cell multi-omics in general, but we really haven't done much in looking at where that RNA is inside a single cell. And so VisGene and some of the other technologies that you mentioned, it's going to be important to start, if we're going to be expecting AI to have predictive biology, we're going to need a better understanding of what does the city look like inside the cell that these RNAs are moving in and out of, and how, to your point just a little bit ago, you said when there was more mRNA there, you were able to get more like IL-2 or IL-7. We need to understand what that looks like, what's helping traffic down there. So it's really, we've done a lot to get to this point, but we have so much more to do before we're having AI predict what's going to happen next. So that's not really a question, but if you have a comment.
No, but actually it's a topic of interest for my old lab, and it's something they've been working on to visualize, what type of proteins are attached to these mRNA species. Can you do co-localization? And for that, of course, flow cytometry is not that well suited. Imaging cytometry, the resolution is mostly poor. So they've been relying on supermicroscopy, and the results are really interesting. Look up my old lab, the lab of Monica Wilkerson. You'll find some really cool work on that.
Yeah, we'll do that. Great job again, Julian. Thank you so much.
One
Oh,
question.
good.
Sorry. We have one question from YouTube. Julian, they would like to know if you can briefly explain the differences between things like prime flow and fish flow.
Yeah, so Prime Flow is a commercially available. I think it's Thermo Fisher by now. It used to be Invitrogen, something like that. And the between brackets difference is the way that they build up the fluorescent signal. So most of the homebrew assays, they're just 20, 30, 40 primer sets that are specific for, let's go with IL-2, and they're spaced out across the mRNA species. They're labeled directly, so you're picking up a one-to-one signal ratio with Flowfish. With PrimeFlow, there are little adapters attached to the probes, and you build out a tree that supposedly amplifies the signal and should result in a better signal-to-noise ratio. In my experience, that hasn't always been the case, but it can be a way to detect very low-level transcripts. So that's the difference between those two types of detection techniques.
All right. And with that, we'll move on to our next speaker. Thanks again, Julian.
You're welcome.
So first, I'm going to read you just a short snippet about Dr. Mead, and then I'm going to, do my thing and tell you why I invited him. Dr. Mead is a principal scientist and director of both the Translational Immunopathology Lab and the Clinical Immunopathology Lab at St. Jude's. So these labs provide support for the research aims at St. Jude's-led clinical trials. Dr. Mead is a licensed medical laboratory supervisor, and he serves as the technical director of that clinical flow lab. He received his undergraduate degrees in biochemistry from Massey University, New Zealand, and his postdoc training was at Harvard Medical School in the Department of Hematopathology and Oncology. So as an independent NIH-funded investigator in the Department of Pathology, Dr. Mead's research interests are focused on the development of hematopoietic system during early embryonic development. For more than a decade, Dr. Mead has been the director of that lab, where his research interests have focused on immunophenotyping and minimal residual disease detection in pediatric malignancies. Most recently, these efforts have been directed towards developing high-parameter spectral panels for diagnosis and treatment follow-up of pediatric leukemias in the clinical laboratory setting. So the first time I saw the initial work from Dr. Mead's lab was, I think, 2 years ago at ICCS. And I know not everyone in this room gets to go to ICCS, but it was on Sony's new instrument, the ID7000. And they've put significant work into developing these panels. And it really actually shows to the support of guys like Jeffrey Clapper out of Sony's technical application specialists. And I was just very impressed, and we've continued at ICCS to build on that. SciTech has shown a nice panel that they're building. We're definitely seeing a shift in the clinical world to adopting spectral flow cytometry. My main reason for asking Paul, besides the talk is fantastic, is I want you to see the level of detail that they put into it, and really looking at this as a gold standard, because as we move into clinical spectral flow cytometry, it's going to be crucial that we're unmixing appropriately and that we're really doing right by the patient. We're not telling them to go home and eat a banana, and these tools can be complex to implement. So with that, Dr. Mead.
Do I need to plug? Oh, goodness. All right. Oh, thank you very much for that, incredibly generous introduction. I was actually sitting here wondering who she was talking about. Okay, so as mentioned, I'm going to talk about our efforts to bring spectral flow cytometry into the clinical environment. And oh, why is that not working? Oh, some reason this seems to have frozen up.
Or maybe you have to click.
It seems. Oh, here we go. Thank you. Oh, there we go. Thank you, Rui. One of the many times I've had to thank him. Okay. My first up, unfortunately, I've got nothing to declare. I have no conflicts of interest. So anyone out there, but yeah, just all jokes aside. So today I'm going to briefly outline our talk is going to be like this. So first up, I'm going to show you what our current workflow is like in the St. Jude Clinical Immunopathology Lab for a newly diagnosed either new leukemia or relapse that would come into the clinic. And I think that will give you a very clear idea why spectral flow cytometry was so attractive to us and why we decided to embark on this very long and difficult journey. I'll give you a very brief overview of spectral flow. Probably this audience doesn't need that at all. But like Peter said yesterday, I put so much effort into the animations, you're going to see it anyway. And then I'm going to talk a little bit about our 38-color immunophenotyping panel design. I'm going to really focus just on one laser in particular, on the red laser, and you'll see why in a minute. And we're going to wrap it up with our efforts to get this panel validated in the clinical lab. I'm going to just touch on a few points of our validation plan. But we've now had this active in the lab for almost 6 months, and it's been our upfront immunophenotyping tool, and we've been reporting on this in the clinical lab for 5 or 6 months now, and it's working really nicely. So there are going to be 2 heroes in our story today, and the very first is a really talented physician scientist in my translational lab, which is where we do all of the assay design and also clinical trial work that we do for St. Jude-led clinical trials that need flow studies. And this is Peijing Liu, and he has done all of the development work for the development of the 38-color immunophenotyping panel that we'll display today. But before we get into that, I just want to talk a little bit about our current workflow in the clinical immunopathology lab. So if a patient comes in with a suspected leukemia, we draw a sample, and on day 1, we will undergo an extensive immunophenotyping panel. And really the reason for that is to identify the immunophenotype of the lineage assignment for that suspected leukemia. And then also on that same day, we will also run if it turns out to be a B or T or AML leukemia, we will also run our MRD panel in order to get a baseline for the MRD analysis for later on and for treatment follow-up. So at St. Jude, our immunophenotyping panel is quite extensive. It consists of 13 tubes on the 8-color Canto 2. And it consists of over 50 markers. We have 46 surface markers and 4 intracellular markers spread out over 13 tubes. And this is performed on every new leukemia that comes through. And again, for the last 12 years or more, this has been done on the workhorses in our lab, in the clinical lab, the BD Canto 2, the 3 laser 8-color models. We have 5 of these instruments, and they run continuously all day processing samples. Upon analysis of that immunophenotyping data, we get a lineage assignment after the hematopathologist has signed off on what lineage it is. We will then go on to determine which MRD panel needs to be run. So in this case, for example, if it was a B MRD, B-ALL leukemia, then we would run the B MRD diagnostic panel consisting of another 9 tubes of antibodies. And all of our MRD panels are made up around backbones. So in the B panel, we have a backbone of CD45, CD19, 22, CD10, and 34. And the idea of the backbone for each of these different panels is to identify the blasts. And then we have these, what we call these add-on markers, excuse me. We have these add-on markers in the remaining 3 channels that we use to identify a leukemia-associated immunophenotype, or we're looking for markers that show us a difference from normal expression in a normal developing hematogone or regenerating B cell. So just to give you an example of what this would look like, here's a diagnostic sample of an acute B leukemia, and using these backbone markers, we can identify, for example, here we have these CD45 DIM 19 positive cells that overexpress CD34 and also have high levels of CD10, so clearly a blast population. Oops, going the wrong way. So then we use our add-on markers to be able to identify a leukemia-associated immunophenotype. And this one would be relatively simple. We can see overexpression of CD73 and underexpression of CD38. Now, all of these other gates that you can see here, we took a whole bunch of normal regenerating marrows and ran them on the panel. And so these show the limits of where the normal hematogones or regenerating B cells would reside. So anything outside those gates would be different from normal or indicate our leukemia-associated immunophenotype that we could follow during different time points. So for this patient, we would use a combination of 38, 73, and 20. That would be our primary tube to follow this leukemia. But we always choose at least 2 other tubes as well to be able to, just in case there's phenotypic drift or whatever during treatment, because we know, for example, like CD73 goes down with chemotherapy, so it may not be as useful later on. But you can see on day 1, this is what the workflow looks like, and it's really extensive. We've run over 50 markers here. We've added another 3 markers that weren't already run in the immunophenotyping tube. A total of around 22 different tubes shown here. And I'll show you one other tube that we also run, all to get a patient-specific follow-up panel that we would run. So you can imagine, not only is this a lot of work, but there's also a lot of behind-the-scenes sort of pre-analytic work. So anytime this patient comes back to the clinic for an MRD follow-up, the techs need to realize, okay, we've got this patient, this is a specific panel that needs to be run. So there's an awful lot of pre-analytical work that needs to be done in addition to all of this wet work on the bench and the analysis. To give you an idea of our workflow or volume, we see around 300 new leukemias a year, and we run approximately 1,600 to 2,000 follow-ups every year. So a lot of work, obviously, for our immunopathology lab, and a lot of, obviously a lot of pre-analytic work trying to identify and make sure that the right patient's sample gets run on the right tubes for the right panel for each time point. So this is really what attracted us to using the ID 7000. And so I'm just going to skim over this really quickly because this audience does not need any sort of primers on Spectral. But again, as I said, I spent a long time on the animation. So on the ID 7000, of course, is that the big difference from a normal cytometer is that for each laser line, we measure the entire spectrum of the output from that laser line using a series of PMTs. And we measure the entire spectrum from about 375 nanometers out to 840 nanometers. And really the advantage, we use the 6 laser versions of the ID 7000. And so in total, we have in excess of 182 different detectors that are measuring fluorescence output from these detectors from each cell. So for example, if we're measuring something like PE or an individual cell, we end up with this measured vector. This is essentially the shape of PE. So you can consider this the overall combined spectral shape of the PE fluorochrome, which is invariant. Now, this is quite different to what we would do on the Canto 2. So if we look at the same sort of running PE on the Canto 2, we essentially throw out all of that other information and we focus in using a series of filters and mirrors to look at a single PMT to count photons around the emission maxima of the blue laser. So in comparison with the Canto 2, we have one data point, whereas in the Spectral, we have 182 fluorescent data points. Now, because of this, we have to no longer be able to use simple subtractive compensation. And as Peter Mage and others told us yesterday, we have to use linear algebra terms such as a weighted least square, which is used on the ID 7000. And this is really old math, right? So this was invented back in the 1840s by Gauss. And it's been used extensively in earth science and astronomy for many decades, but it's relatively new to cytometry. And essentially what it does is we take the shape of those measured vectors for each of our different cells and it says we have what combination and how much of the shapes that are represented by our single color controls or our reference spectra give us the best fit for that line that we generated, that measured vector. And that turns out to be our parametric data, and then we can plot that as histograms or dot plots using as a standard FCS file. Now, obviously, the real advantage of Spectral is that the fluorochrome is excited by all laser lines, and we collect data from every detector all at once. And many fluorochromes are excited by more than one laser. So we use all of this additive power of all of the different data sets. So we combine all of this and it adds to our ability to do the unmixing. Now, nothing comes for free in this world. And just like in conventional flow cytometry and spectral, we have to deal with problems like spectral spillover. Now, there's no physical way around this. So the only way we thought to do this, and again, we did this around 4 years ago, and we wish we had some of the tools that were discussed yesterday to be able to identify hotspots in our panel where we should really have tried to move things around. Instead, we did it the old-fashioned way and just went to the catalog and bought everything that we could buy and then just pipetted things at random. That works as well, but obviously now I would not hesitate to emphasize, use the tools that are available for the bioinformatics is making this whole process so much easier. So anyway, the way we went around it was that we used a spread matrix again, and we took 45 different fluorochromes that at the time, again, this is like 4 years ago, then we started this project, and we said, okay, where are all of the hot spots, or where is the spread occurring? And I'm a sort of simple kind of guy. We just decided that anywhere where there's a lot of red, which means a lot of spread, we'll just forget about that one. And so we took that out of our panel. It was not very subtle. So we ended up with about 38 colors that we decided that we could use that would be a reasonable number to be able to try and squish that entire 50 color panel that we run on the Canto 2. We'd try and squish as much of that into a 38 color panel on the ID 7000. Now, the other limitation that we did, and again, remember this was around 4 years ago, it's not such a big deal now, but at the time we decided that we will only use commercially available antibodies. We're not going to do custom conjugates because obviously the idea was to bring this into the clinical lab. We wanted to make sure wherever possible we could use ASR reagents. If not, we didn't want to have to do individual batches that were made specifically for us. We wanted to just use commercially available conjugates. And so then we came up with this panel that we sort of fitted into this overall panel to generate a 38 color immunophenotyping panel with 31 surface markers and 5 intracellular markers. And we also included 2 dyes, one a live dead marker and a nucleated dye, and we're going to talk about those in some detail in a minute. But really, the overall aim here was to generate a palette of colors that we could use for future design of other panels, right? So not only were we designing this 38 color panel, but we knew we wanted to also make MRD panels for Bs, Ts, and AMLs. And so we wanted to generate this sort of palette that we could then fit into our overall workflow so that we wouldn't have to redesign the wheel, as it were, each time we made a new panel. And you'll see this in a few slides. Now, I'm not going to go into too much detail about panel design here. I'm just really going to focus just on the red laser and those dyes that we added. So we decided to include into our panel, and every panel that we design, we include a nucleated marker, and we use DRAC5 for that. We also included a live dead. We use zombie NR, which is quite a common thing. Because we're a hematopoietic, nearly everything we're interested in is going to have CD45, either at high levels or absent, but CD45 is really important in our early gating scheme. So we decided to keep these sort of what we call kind of our housekeeping part of our panel down on the red laser, because it's the longest wavelength, lowest energy laser, and there's less sort of feedback of those back into those higher energy lasers where we could keep it those channels open for our more specific markers, as it were. Everyone says, why do I hate the red laser? I actually love the red laser, and it's been really useful for us to be able to include this. Now, you probably think, now, why do you do that? Why go to the problem of adding a nucleated marker as well? So this comes back to yet another tube that we run on the Canto 2. Anytime we do an MRD analysis, we also run. So we'll run our MRD panel on our cocktail and our added markers, but at the same time, from the same sample, we will also run a Cyto13 tube. And the reason for that is to help us calculate the denominator for our MRD calculation. The numerator is relatively simple. You gate on blasts, and that's the number of blasts that you have. But what do you divide it by? And we use this correction factor. I'll show you the math here. We don't really need to worry about this, but essentially it allows us to convert the total number of events that we record into mononuclear cells that are present in that sample. So we wanted to include the sort of correction factor into every panel that we designed, although we don't need this essentially for the immunophenotyping. For all of our MRD panels, we would want to include this correction factor so that we have a direct calculation for our MRD. Again, that denominator is as important as that numerator when you're trying to determine MRD calculations, number of blasts divided by what. So that's why we put that and spent so much effort into designing the panel around that. So there we have our panel. So then Peijing in my lab, in my translational lab, set about pipetting many dozens of different iterations of this panel and running samples. He ran hundreds of samples. It may look rather simple, but our most complex antigen to fit into this panel turned out to be CD33. We knew there were certain characteristics that we would need for our AML panel. There was CD33 is really important for our AML MRDs, and it sits on the backbone of that Canto 2 panel. So we knew the characteristics of 33 expression that we wanted to mimic on the spectral. So we went through 16 different iterations to find a fluorochrome home for CD33 that we knew would work well. So that just gives you an idea of some of the numbers of different combinations of antigen combinations that we went through. But again, nowadays with these new tools that are available, I think it would be much simpler and a lot less expensive to be able to do that using some sort of computational efforts up front. So again, we Peijing ran these and calculated the measured vector for every cell and compared them to our spectral references or our single color controls. And then we can collect the omega hat, the estimate of the combination of those different individual single colors. And then we can plot those as dot plots or anything. We're cytometrists, we love our plots. And early on in our gating strategy, we included, so we gate on a scatter gate with forward and side scatter. We get rid of doublets using a doublet discriminator. And then we also then use our zombie NIR, our live dead marker versus our nucleated to be able to gate on this population of live nucleated cells, eliminating both dead cells and cellular debris. You might see in a few slides, my Peijing in my lab likes to call this the Mead gate. I call it sometimes he changes it backwards and forwards. So if you see that, that's what it is. It's very, very sweet of him, but yeah, a little bit embarrassing. So if we now look at the spectral quality of this, so by running this live nucleated cell gate very early in our gating strategy, we significantly clean up our spectral profiles of our cells. So here are the dead cells on the top, and you can see now that we've got this live nucleated cell events, we can see much more patterns arising in our spectral data. So then, of course, we can just gate from there and then start gating with our fluorescent markers, and we can gate on, say, CD45. You can see nice lymphocytes, monocytes, and granulocytes. And then we can start including some of our specific markers for like B cells, different T cell, and then also myeloid lineages. So the panels seem to be working fine for distinguishing all of these different cell populations. Now, just as a bit of an aside, I threw in some other slides because I'd really like some feedback from people later on. So another reason that we added our DRAC5 into there, or another sort of happy accident, I guess you would say, was part of the thoughts we were having around our minimal residual disease panels, where we have to collect really huge number of cells to be able to do these calculations. Our current MRDs, say for B or T MRDs, our sensitivity on the flow on the Canto is down to 0.01%. So if we want to go down to that level or even lower on the spectral, obviously we have to record vast numbers of cells. And that sort of leads us to the real spectral elephant in the room. I'm from New Zealand, and we say a taniwha in the hot tub. It's basically the same thing, and this is data management. So I did a bit of back over the envelope math. Actually, if you look at the math, it's probably a back of a cocktail napkin. Anyway, don't disregard the math. Just suffice to say that the data files get massive, obviously, very quickly because of the number of simply the number of channels that we're recording for each event. So we need a strategy to manage the data. So being a cytometrist, one of the first things you think about is setting thresholds. So the first threshold that we would set would be forward and side scatter. Now, as we heard really elegantly yesterday, you've got to be very careful about your forward side scatter because it's not a direct indication of the size of your cell. The other thing that we need to keep in mind is that some blasts can be very small. So we wouldn't want to set our threshold too high and throw out any of our blast population. But we can we can manage our forward scatter threshold to be able to at least get rid of some of our debris. So then we thought about our gating strategy and some of the early gates that we are using. And so in this case, and David mentioned yesterday that it should be reported to Cell Protected Services. We took a sample that had been left out on the bench for about 4 or 5 days. It was an EDTA, peripheral blood tube. We just left it over the weekend, left it Monday, Tuesday, I think Wednesday that we came back, and we actually stained it up. And we wanted to find a really poor quality sample. And so we ran it on we did a bulk lysis and then ran it on sample. And you can see only out of the 50,000 events that we recorded, we only had about 10,000 events available in our live nucleated cell gate, and there's tons of debris. And we just randomly gated on a few populations. We've got some B cells, some T cells, CD4 T cells, and some monocytes. And we calculated their. But then if we go back and then look at that scatter plot and look at the spectral profile of that, you can see it looks kind of ugly, right? You can see this long line of what essentially is going to be all of that debris. And fair enough, if we look then only focusing on those live nucleated cells, the spectral quality of that looks really quite nice. And you can actually see quite a lot of information is retained, even though from this really old and sad sample that we started with. And then conversely, if we gate on that debris gate, of course, what we see is just this big line of junk down the bottom that's sort of contaminating our nice spectral profiles. So then if we then look at what information, where are we gathering that information? It's from the DRAC5 channel, and that's on the red laser. So then if we look at that spectra, we can see the absorbance maxima for DRAC5 happens to occur on the red laser on PMT number 21, and it measures between 690 nanometers to about 700 nanometers. So this 10 nanometer window is where that DRAC5 emission maxima sits. Now, we are only interested in nucleated cells in our studies, so we thought we could also then add in a secondary threshold only on that single PMT out of the 182. If we set a threshold of, say, 10% on that, can we eliminate all of the debris from our data set? And it's exactly what we see. So if we run it with the threshold, the secondary threshold set at 0, and then our secondary threshold set at 10%. Peijing loves doing lots of runs, so he did it at various intervals, and we found the sweet spot was around, oh, I'm so sorry. Found the sweet spot was around 10%. So then if we then run this and then do the data analysis again, just randomly gating on those same populations, we can see that they're exactly the same. So we haven't lost any of our data that we're interested in, the live nucleated cells. The proportions are still the same, but now we've got at least out of our 50,000 total events, now we've got 30-odd thousand of our live nucleated cells out of that really disgusting sample that we ran compared with only. So we've got at least a 3-fold increase in the number of actual cells that we care about. And then, so in practice, what we do, we always run a small event tube, so 50,000 total events. And then for our mini event tube, for our MRDs, we run using that secondary threshold in place. Okay, so back to our panel design. Sorry for that sort of sidestep, as it were. Now, all cytometrists, as we heard about really nicely yesterday from Anath's talk, these autofluorescents, and really one of the real advantages of spectral is be able to identify and manage autofluorescence. And again, as we saw yesterday, we can gate on those and define their emission spectra profiles, and we can add those to our unmixing matrices, and we can do the unmixing with and without the autofluorescence, and it significantly improves the overall pattern of our unmixing. And unsurprisingly, it dramatically improves our unmixing across the entire panel, because now those photons that are due to autofluorescence are put in the right place, they aren't randomly spread out and causing errors across the entire matrix. Now, we can export these files as FCS files, and we can use a variety of different third-party softwares to do the analysis. One of my favorite ones is Infinisight, because it handles the large data sets really well. And also we can see everything all at once on a single page. And as we learned yesterday, the principal component analysis is a linear projection. And so, for example, in this BALL sample, for example, you can see the red blasts, and they segregate most closely with the normal B cells, because they're most closely like normal B cells than any other population. So at the end of this point, Peijing had done many samples and lots of different combinations. We ended up with a 38 color panel. We had to draw a line in the sand, as it were. We knew it wasn't perfect, but perfection is the enemy of progress, as Winston Churchill said. So we decided it's good enough to move forward. We couldn't just keep doing iterations until it was perfect. We had to then do the next step, otherwise we would never get anywhere. So at this point, Peijing had run a total of around 82 different samples that had been run on the clinical lab and taken over to my translational lab. And so we've done, like, as you can see the numbers, lots of different samples. We had 100% concordance with the output, which is it a BT or AML? And it's a really pretty low bar, but we had 100% concordance between the clinical data and the data that he generated on the new translational panel. So the next step, we cleared out an entire bay in the clinical lab. I'm also technical director of the clinical flow. If you work for a non-profit, you end up with multiple jobs. And so we cleared out and we bought 2 new ID7000s. We got nice big 50-inch screens, and we called them Isaac and Newton. because I don't get out much. And so then comes the next hero of our story, Laura Key, a clinical translational scientist now in the clinical flow group. And she has done an absolutely stellar amount of work to be able to bring the panel that Beijing had put together and bring it into the clinical lab. And this entire process took around 18 months, which is incredibly quick. So big kudos. So just to give you an idea, this is only part of the checklist for our validation plan. If you, I think one of those lines that you really can't read and you really don't need to, says write new SOPs. We have over 300 pages of new and revised SOPs, training documents, and yeah, whoever the CAP inspector who comes and looks at that first time has got their work cut out for them. But at the basis of this entire validation plan was we wanted to run and compare to the Canto 2 data on the same day or the next day if possible. At least we wanted to do 20 Bs, 20 Ts, and 20 AMLs. So a data set would be 60 comparisons between the clinical lab and the Canto 2 and the ID7000s in the clinical lab. So Beijing handed off the panel to Laura. The first thing she did was go back and retitrate all the antibodies, as one would do. Now, it's really important to note, in the initial studies that Beijing did, he pipetted every single one of those samples by hand. There were no cocktailing. It was one antibody at a time for all 38, for all of those many, many different iterations. Now, that doesn't really work well in the clinical lab, and we decided that we really need to do some cocktailing. Making a cocktail with 36 antibodies would, one, be terrifying. If you made one mistake, it would cost tens of thousands of dollars. And the other thing, too, was that we knew that this was not going to be the final iteration of our panel, and that we wanted to make changes. And so it was much easier to make a cocktail of cocktails. So we made 6 cocktails of 6 antibodies, and then we cocktailed the cocktails together and then added to the, The other really important thing is that, generally when you're making cocktails, there's certain rules about what you put together and what not. We sort of didn't really look at that. We decided really just to maximize the efficiency for us to be able to verify the cocktails. So we needed to use cell lines and spike-in samples to be able to make sure that the cocktails were working as predicted. So our cocktails were actually designed around making it more efficient for the techs to be able to verify them. And it seemed to work out well. We got quite lucky. And the next thing, we did some cross-platform assays between the Canto 2 data. So we get a sample into the lab, run it on the Canto 2 data, get reported, go into the medical record, would run the same sample on the ID7000 and then compare the data for both. So we ended up with this cross-platform worksheet. On one side, we have the Canto 2. On the other side, we have the ID7000. And from the medical record, we would pull the data for each of the data sets for each of the different fluorochromes. And then, obviously, we would compare it on the other side to the ID7000. Then we would have 2 different types of flags or discrepancy. For us, a flag would be if it would be both positive, but one was dim and one was moderate. Now, you can imagine going from an 8 color to a 38 color. It would be virtually impossible to match all the fluorochrome brightnesses. We did our best, but there's going to be cases where one's going to be brighter than the other one, and et cetera, right? More importantly, we wanted to look at discrepancies. And this is where one was positive and one was negative. We really wanted to know and make sure that we identified those early on. And we did, right? So in the first batch of cocktails, so for the first 25 patients, we identified particularly there were 8 for the CD11B on the BV421 on the Canto and BV785 on the Spectral. There were 8 times when the Canto was positive and the ID7000 was negative. Now, this would make no difference to the caller, whether it was a B, a T, or an AML. It would make no difference to the potential treatment plan for that kid. But obviously, we need to address this. This is not right. So what we did, we went back and looked at our titrations for this. So as one does, Laura did these titrations individually, and you plot out your stain index and everything, and you get a beautiful plot, and you look for the breakpoint, and you're like, that's the perfect spot for my antibody. But it turns out that, so then we went back, made the cocktail that 11B lives in without 11B, and then titrated in the context of the entire cocktail. And you can see that it's quite a difference, right? So if we just add the amount that we would do individually, well, it's really not enough, right? And so in the context of the cocktail, we needed more. And so there were a few antibodies where we saw that they caused discrepancies, and almost always it was a case where we'd undershot how much antibody we should actually use. And in one case for MPO, we had to do it in the context of the entire panel to be able to make sure that the level of MPO expression was where the hematopathologists were used to seeing MPO sit for normal granulocyte or monocyte populations. So, so we went through a lot of trouble to make sure it was also looking similar to what they've always been looking at for the last 12 or 14 years. Now, the other thing that we found, and this is probably no surprise to anyone, but it was to me, was that our Q dots really don't like being in cocktails at all. And we found that we were really rapidly losing. See, we had our Q dot was the only thing on the 320 nanometer laser, and we were rapidly losing signal. So 2 weeks of age of our cocktail, almost the CD2 population is gone, and by 5 weeks it was pretty much non-existent. So what we decided for this one was to actually make the cocktail without it and just add CD2 Q dot as a drop-in. And then we do that, it looks great, right? But it looks like a dog's breakfast if you put it in the cocktail. So then Laura went through and did, towards the end of the validation plan, we had 82 different samples that had been run on the Canto 2 and also on the ID 7000. And, and a good mixture, almost reaching our targets of what we wanted, right? Okay. And again, we had 100% concordance. All the Bs were Bs, all the Ts were Ts. So then, just and the final point, we just talk about sort of our workflow for the spectral and the clinical flow lab. So we run our, we stain and run our sample on the ID 7000. We've defined the autofluorescence, and this was talked about yesterday really nicely by Anna and some of the other speakers. And then we do our unmixing adjustment. We'll talk about that in just really, really briefly. But really what I want to point out is this bottom part here. And with all of this validation going on with validating a brand new instrument, a brand new panel, we wanted to keep part of that workflow the same, right? Now, on top of this, at the same time that we were doing this, we were moving our medical record from Cerner to Epic. So the last thing we wanted to do was bring in yet another third-party software that we'd have to validate as well. So we wanted to try and keep this part of export the FCS file. This is what we did on the Canto 2 for many years. We'd take it into FCS Express 6, and through our middleware called Data Innovations, we could take those tokens from FCS Express and put it directly into the medical record. There was no transcription needed, right? It goes straight from one software to the next. So we wanted to do that with our workflow for the ID 7000. Turns out FCS Express 6 didn't work at all, but the new version 7 worked perfectly, and we're able to use FCS Express 7 through our middleware. Of course, we had to validate that it was working and run trials, make sure that the right tokens and everything were going across, but this was able to export that into Epic just fine. I'm almost done. So just give you a really quick rundown of our unmixing. This was by far the most difficult part of our validation, was to generate an unmixing strategy or SOP so that no matter which tech in the lab did the unmixing, that they would always look the same, right? We didn't want any sort of variation from person to person. So we came up with a very strict set of rules for unmixing adjustments. We always gate 2 autofluorescent populations. The reason that sometimes we need sometimes 2 very distinct autofluorescent populations, but FCS Express is always expecting the same number of channels, and so you can't have sort of like 1 autofluorescence in one sample and then 2 in the other. So we decided always just to gate 2 autofluorescent populations. We manually check the unmixing. We always start with the autofluorescence channels first, then the red laser with that DRAC, which is usually the most problematic of the guys. We only adjust negative populations, and hopefully I can show you that real quick. We always start with the most impacted fluorochrome first, and then we save that new unmixing profile as a new setting. So just really quickly, we go to the autofluorescence, and sort of what we look for is things like that, where we have this sort of negatively bent. We call them southern hemisphere bananas, because in the Coriolis effect makes bananas grow the opposite way in New Zealand and Australia. And so you have this negative banana, and so we want to straighten it. But you notice the guy right above it, it's not as bad as that one, but it's sort of negatively bent anyway. This is what we mean by start with the most impacted one first, because if we fix this one is automatically fixed. So you can imagine if you started with that one and you fixed it, and by the time you did the worst one, then you'd have to go back, and it turns into a nightmare. You'd never know where to end. Okay, so and then we just check the unmixing settings to make sure that we haven't gone too far. There's information on the worksheet to tell you, have you done something horrendously wrong? And if you haven't, then you can go ahead and export your FCS file. We import that into FCS Express 7, and we can generate our tokens. So here's an example of a relapse BALL that we ran on the Canto 2, and this is the FCS Express 6 worksheet for that. And you can see here's our interest gate of our blast, and it looks a little bit ugly. These are obviously dead cells here, but you can see these red cells represent the relapsed BALL. And here is the same sample run on the ID 7000, and you can see it looks a lot cleaner, right? Because of our live dead nucleated markers, we are able to identify just the live nucleated blast cells, and we get a much better population. So then in FCS Express 7, we're able to then do all the plots, and we can see for this one, we've got a subset of CD10 positivity. And then we just use a variety of histograms, and then we just sort of compare it to the isotype control, and the techs go through and score each of the markers. Then a hempath would come along, whoever's on service that week, and check it, and then that would automatically get exported to the medical record. These become the tokens for export into the medical record, so there's no transcription. So what about sort of just in the last minute? So we're in the final process of obviously doing our St. Jude panel IP38. We've moved some of those problematic. We've moved that CD2 off the Q dot and put it on Real Blue 613, and it works really nicely there, and we don't have those issues with cocktail stability. We're looking into robotic systems for doing cocktailing. Obviously, the less pipetting steps we can do, the better, and also potentially staining. We're also looking at, as we heard really nicely yesterday, looking at AI tools for data analysis. One of the things I'm really interested in is using AI tools to be able to help us verify or validate our cocktails from month to month, to make sure that from month to month, are we getting the same type of UMAP from that cocktail as we did from last month's cocktail or whatever. Okay, and it turns out we probably need to bank a lot of some apheresis rings, so we have the same sample that we can measure month to month across that. And then while Laura's been doing that 18 months of getting this activated paging back in my translational lab, has been working hard on the spectral panels. So just really quickly looking at some of these AI tools, turns out that this live nucleated cell gate is a great way of cleaning up our data. For us, it seems to be equivalent to Peacock. People are probably laughing at me, but if we gate on that live nucleated cell gate, if we then run Peacock, we lose no other events. So it seems to be as good as doing that. It generates nice flow sim data, and the UMAPs from even 50,000 from a normal peripheral blood, we find all of the populations that you would expect to see in a normal human peripheral blood. So then just really quickly, the MRD spectral panels, obviously want to move our entire workflow to all our high content flow to MRD, to the spectral. We used our palette of colors again. Beijing has developed a 39 color T-ALL MRD panel. And this just shows for the same patient on both the Canto 2 and the ID 7000, they look the same. Likewise, he's generated a 40 color B-ALL MRD panel. And again, looking at, it looks exactly the same on the Canto 2 and on the ID 7000. We're in the process of validating both of these panels in the clinical flow lab at the moment. Okay, so let's get back to our current or existing workflow. All of these tubes, all of this pre-analytical work. And really what we want to do in the future is to move this, and we're this part of the puzzle has been solved. We've gone from that really complex immunophenotyping panel to the single tube spectral panel. We've developed the panel. So you can imagine if this child has a TMRD, a T-ALL as it were, anytime that they come in, and no matter where there is, that we no longer have to worry about assigning the right panel to the right. If it's a T, it always gets the T-MRD panel at every time point. And so there's all that pre-analytic work is gone as well, because if it's a T, it would automatically just get that one panel. It would have all of those other markers that we would look at as well. So finally, I just want to acknowledge all of the really talented, in the translational lab, I pointed out the work done by Peijing Liu on the development of this panel. Mo Mu works on mainly BH3 profiling, and Zhao Hong works on CSF and also some quantitative flow. Really, none of this work would be possible without our really talented group of hematopathologists. We're led by Jeff Klo, our division director, Priya Kumar, our medical director, and Marsa, Gabriella, and Yen round out our really talented team of hematopathologists. A big shout out to Jeff Clapper from Sony Biotechnology. And really the real unsung heroes of all of this are the clinical lab techs and people who do all of this work over in the clinical lab, led really well by our manager, Christy Embry, our two scientists, Laura Key, who I showed you did all of that validation work, was a real tour de force. We've recently been joined by Stacey Willard, and she's going to be doing a lot of the data analysis. Our supervisor, Celine, Lisa, Chris, Pam, Rath, and Robin are all of our med techs. And with that, I'll take any questions. And I put a picture of my cat, so you won't ask any tough questions. Uh-oh. Or if I went a little bit long.
That was a really cool talk. I'll ask my question first because I'm standing here in the room. You've got a ton of data now, like a bunch of markers on all of these cells beyond what you would normally have from the Canto 2. So aside from just trying to like automate data analysis, will you also look to do some discovery for maybe unique populations that aren't typically seen?
Absolutely, right. And so, even with that 30, even with the Canto 2 data, we have, years of data from, running those 13 tube panels, but it's better now because it's all in one tube with the 38 color. Yeah, that is definitely our goal. And it's one of the things that, we've hired Stacey into our group as part of our data analysis. And hopefully, we really want to take advantage because there's just a wealth of data. So not only in the flow, but being able to correlate that with all the molecular. So we for every patient, they'll do RNA-seq, whole genome, whole exome as well, and being able to combine all of the genetic and molecular data with the flow data and any other metadata, as it were. It's an absolute goldmine, I think, for future analysis. Yeah.
Very cool. Okay, we've got a few online as well. So a couple people interested in the cocktailing, how you're doing it. So one person is asking, how long will you use the cocktails for? How long do they last? Which I guess we know the Q dots don't last long, but other stuff. And then another person asking, how are you preventing interaction between antibodies in those cocktails?
Oh, yeah, really good questions. So we started out with using making cocktail every month. So 5 weeks, I guess, 4 to 5 weeks. One, just because we knew that, there's a lot of issues. I think we could probably take that out to 2 months without, so we do cocktail stability assays. CAP ensures that if you do anything beyond 1 month, you have to revalidate at 1 month. So that was kind of why we decided first to do it 1 month, because if we have to revalidate anyway, we may as well make a new cocktail. It turns out that's quite a lot of work. So we prepared everything by hand into the cocktails at the moment, if that's what they're asking. We haven't got a robotic system. The robotic system I really liked. They went out of business. They got bought by another company who then killed them. It was an instrumental robot. But anyway, so we're still in the market for looking for robotic systems. So if anyone's got some ideas, that'd be great. But yeah, so when we make our cocktails, we throw in every single blocking reagent that we can into the actual cocktail. So yeah, that's how we try to get around any interactions. Obviously, the BV buffers and pretty much anything that's on the market, we throw it in there and hope for the best. And so far, from our validation studies, they seem to be fine for that period of time. But yeah, it's definitely a question. Yeah.
There's some more technical questions too. Let's see. Are you using any FMO or FMX tubes? And a question also about voltages, if you keep them the same, but I suppose maybe you don't need to change them from the jump.
Really, really good question. So, yeah. So what was the first one?
FMO.
FMOs. Yeah. When we're doing the panel design, we were using FMOs a little bit, and I know we should be using them more. But just in practice in the clinical lab, to be able to run all of those different FMOs would be impractical. During our design phase, to make sure for certain markers that may have been, were either flagged, or then we would sometimes go back and essentially run FMOs, like on the MPO, for example, that looked fine, but it was not at the level where the HEMPAS we're used to seeing it, so we had to adjust it a little bit. So certainly for those ones, but in general, no. And I see the criticism, but it was just a practical, the ability to make all 38 different combinations and run them to be, part of the real reason for, another reason to do this is that, in pediatrics, you get a very small sample volumes, right? So if we're running that number of tubes, you're wasting lots of cells. So one of the real advantages of Spectral is that we're saving so many cells, right? They can be banked and used for other tests to help, And so the real, that was probably the main driving force why I think our admin people got on board with, spending the amount of money that we needed to do to bring in 3 ID7000s and bring this all online, was really that we'll save sample, and so that sample will be available for other testing modalities down the line. So yeah, really useful way of not answering your question.
It's a yes and a no.
I answered the question I'd like to answer.
I'm going to one quick question here, since MRD is kind of measuring change, right, over time. Have you found any use of the autofluorescence in time on this? Because as we're getting more, you have a 6 laser, the 320 laser, we have basically you're characterizing autofluorescence, which is characterizing cellular change, or at least, so do you see any of the autofluorescent changes in population to help find MRD?
Oh, that's a really good question. No, but we will certainly be looking at that. We're just sort of, at the moment, we've been running just really early time points on our MRDs, just for practical reasons, because we're in the sort of panel design phase of it all. And so we're actually looking for ones where we can obviously identify the blast population, right? And so yeah, it's going to be much more critical as we get lower and lower in our sensitivity to be able to identify those. And whatever parameters work, we'll definitely include those, right? We're not yet, definitely would, particularly after hearing Anna's talk yesterday, we'll definitely include.
Thank you so much. Great talk.
Sure.
Yeah, I'm just wondering, maybe this is a, big step compared to what you have already there. But you were mentioning about the blasts and, debris, etc. Have you thought of the advantage of using imaging, like a spectral imaging flow cytometer for,
Yeah, that'll be brilliant.
right?
Yeah, I think, yeah, that'll be, yeah.
To have the pathologist also confirm,
Yeah,
this is what it is.
exactly. I think the new technologies are only going to add to the,
Right.
most definitely. That's a really good point. I'd love to see what they look like on it.
In the interest of time, we're gonna let you guys go on your break.
Yeah,
and
sorry
go
about
on.
that.
I'm gonna make one small comment while you guys are walking out. You do not have to stay seated to hear this, but I couldn't help but think of NVIDIA has their earnings call today, and their CEO said that if he knew what he was getting into all those years ago, he would have never done what he's done. And I wanted to say, Paul, I don't know if you would have never done what you've done if you would have known, but you're on the journey and
Yeah, I
keep
would've.
going.
Oh, thanks.
Yes, all right. So we're going to welcome everybody back from the break. I know it was shorter than we expected, but we've got 2 more talks and we want to try to finish on time to get you to lunch. So you guys might actually know who Daniel Baker is. You might have heard him on NPR a few months ago sharing how CAR T can now be used outside of cancer in autoimmunity and other diseases. I think one of my big, if you've hung around me very long, bones to pick is that we have not used flow cytometry in autoimmunity enough. Dr. Jorgensen here at MD Anderson, I met him early in my career and he kept telling me, you need to meet Andy Rostrom, you need to meet Andy Rostrom. And when I met Andy Rostrom, he was working with a rheumatologist over in Europe, and they were actually monitoring the CD20 depletion in rheumatoid arthritis and eventually lupus. And they had come up with some ways to predict if there would be relapse. And I think one of the reasons we've made so much progress in especially oncology, but especially lymphoma leukemia, is because we have taken this disease, broken it into its subparts, and then not only characterized them well, but moved forward studying in a personalized way the monitoring, especially with MRD. So saying all that to say that I'm very excited to have Daniel Baker here. He is a student, he's an immunologist, but a PhD student at the University of Penn, training in Dr. Carl June's lab and also with Dr. Zoltan Arani. He focuses on expanding the use of cell therapies beyond cancer into autoimmunity, and you guys are in for a treat. Come on, Daniel.
Good morning to all of you. I'm happy to be here. What I want to do for a little bit is talk about the use of cellular therapy and what that means in the context of flow cytometry, all the things that you think about every day. And mostly the impetus of my talk, I have a couple of theses or points that if you take away, I would be happy with. But the major point here is all the things that we think about from a very technical standpoint, and especially you all, from a very technical standpoint every day, has real meaningful clinical utility. And the field at large is starting to think about this. But one of my theses is we are still underestimating how impactful these therapies will be. So to start out, at least as a trainee in the field, it's easy to think of cellular therapy as an iterative next step. I think most people don't think of cell therapy and think iterative, but we talk about it like a living drug. We try to put it into our existing framework because of the regulatory aspects. But to take a step back and to think where we are, what we've started to do for the first time in medicine in the last decade and a half is take the most fundamental unit of life and use it to treat disease. That's totally and completely different than what medicine has been doing for centuries, millennia, right? You can think about the order of complexity of a small molecule, a biologic, even a gene therapy. All of these are exceptional modalities. And yet, if you just think about it from first principles, if you go back to your intro to biology class, think about the layers of complexity of a cell. And for the first time in the last decade and a half, we've been taking and augmenting cells to treat disease. And so one of the points I think we should take away is that as you think about flow cytometry, one of the reasons why it's so impactful is because we aren't looking at a molecule, we aren't just looking at an interaction, we are trying to leverage a living therapy in order to treat disease. And one point I'll make before transitioning entirely to CAR T cells, because that's my focus, is cellular therapy is not just CAR T cells, even though certainly that's what's gotten the most interest as of late. But there's a whole bunch of cellular therapies that are now entering into the clinical pipeline, whether it's in sickle cell disease, early results in diabetes with iPSCs, whether it's CD34 stem cell transplants. And so I think we're going to look back on the decade that we're in and think that things have changed essentially forever because of the experiments, the clinical trials that are ongoing right now. So one of the cool things is, as we think about CAR T cells, I know probably everyone in this room knows what a CAR T cell is, but it's not really just cell therapy. It's really the integration of cell therapy, gene therapy, and synthetic biology. And one of the really wonderful things is, despite its complexity, because it is complex, it's also super easy to understand from first principles, right? Essentially, the idea was a T cell is extraordinarily powerful at killing bad cells. So could we engineer a receptor that unleashes a T cell against a disease? And the proof case here was cancer. So as I think about a CAR T cell, essentially the outside of the receptor, the chimeric antigen receptor, is built to recognize things. It sees whatever you tell it to see. And then the inside, obviously there's a lot of complexity here, but the inside essentially activates that T cell. It tells that T cell, okay, now we want you to start killing. And a couple of simple points that we've taken away is that engineering CARs into T cells, the synthetic receptor that's never existed before in nature, engineering that gene therapy into a T cell can cure blood cancers, at least in a subset of patients. And there's been a huge amount of focus to apply this throughout oncology. Okay, so I want to take a step back. This is maybe the central question of or the central point or question of my talk, which is, where exactly is the field of cell therapy? If you just stop to think about it. Oh, I should also say, this part, this is more hypothetical, but if you have any questions at any point, you can feel free to interrupt. You don't need to keep your questions till the end. So where exactly do you think the field of cell therapy is? Obviously, because of where I am at Penn, as well as in Carl's lab, you know, we believe the hype of cell therapy. We think it's very cool, very exciting. But very often when I'm going to meetings, especially not cell therapy meetings, people start talking a little bit like, you know, okay, cell therapy, it showed a lot of potential, it showed a lot of promise, but a lot of it has not been realized. So what's going on? And the thesis, provocatively, I want to tell you is I think we are past an inflection point in cell therapy. So very often when you're writing grants or coming to conferences like this, it is very apropos for people to say something like, we are at an inflection point. And that's super captivating because that means, you know, things can either take off from here or they can go very badly. But like we need attention and focus and money. I want to say that the field of cell therapy has passed an inflection point, and I want to try to make the point to you that that's an exceptionally exciting thing. So essentially, I think we are here, and just the two parts of my talk are going to be where we've been, the unlikely path of getting cellular therapies actually into the clinic, and then I want to spend the bulk of my talk talking about where we're going. Okay, so this is controversial. You might disagree with me, but I would tell you if you force me to choose an inflection point for the field of CAR T-cell therapy or cell therapy, I think it would be this patient. A very well-known story, Emily Whitehead, treated at CHOP at the University of Pennsylvania. The first pediatric patient to get CAR T-cells. Several patients had gotten CAR T-cells, this engineered cell therapy, up to this point. We didn't understand a lot about it, but essentially, you know, if you don't know the story, a patient refractory to all treatments had been given months to live, and then was infused with CAR T cells. So her cells were taken out of her body, engineered with this gene therapy, a lentivirus, and then put back into her. And within a couple days, she got exceptionally sick. Carl often says, you know, he's never seen a person be more sick in his life and survive. And so at this point, nobody knew what cytokine release syndrome was. Nobody knew that there were neurotoxicities. And so they saw what you can see in black is this super elevation of IL-6 in her blood. And I'm not going to go into the story because it's not the purpose of this talk, but by happen chance, Carl knew a drug because of his daughter that could treat levels of IL-6. And they put Emily on tocilizumab, and within a few short hours, she was stabilizing, coming out of her coma, her medically induced coma. And as you all know well, Emily, 10 years later, post-given months to live, has been cured. So I think this is the inflection point, and I'm happy to talk about it more maybe offline because it's not the point of my talk. If you think there's a different inflection point, I would likely listen to you and believe you. But I think if things had gone really poorly for Emily, I wonder what would have happened to the field of cell and gene therapy. I think about thousands of patients that have been treated at this point, trillions of cells that have gone into patients. What would have happened if Emily hadn't survived? So I think this is the inflection point. And so to just very quickly summarize where we are in cancer, there are multiple FDA approvals now for a variety of blood cancers: leukemia, lymphoma, multiple myeloma. We know that CAR T cells can be curative. They can cause durable remissions, and at least in a subset of patients, 10 years out, a decade out, we can say that some of these patients have been cured. Again, tens of thousands of patients between academic trials, commercial trials, and then public deployment where CAR Ts have actually made it into patients. I haven't talked about it extensively, but there are real toxicities that we didn't realize was going to be an issue, CRS, ICANS. And we've learned how to, or we're starting to learn, and we are learning how to manage these toxicities. And there's a huge amount of work and innovation to try and apply this same therapy throughout cancer. And that's what I think has been disappointing and difficult, is as soon as those initial trials happened, and we saw how exceptionally powerful CAR T cells could be, the question was, okay, the same strategy should be able to be used for lots of cancers. So what's actually going on? In the decades since this has come online, it's been very, very difficult. What is interesting is in the last just few years, it is now clear that CAR T cells are going to start working in solid cancers. Whether you think about some of the Claudin-6 trials where people are seeing real responses, there's been a smattering of glioblastoma trials that have been published in the last 2 years. It is clear to me that CAR T cells will work in solid cancers, but there's a huge amount of innovation that is required in order to get that to work the same way that it's working in leukemias and lymphomas. But what I want to spend most of my talk trying to pitch to you or have you consider is this, is could this same strategy be used beyond cancer? Right? There's been a huge amount of focus, both from academia, industry, venture capital, in getting this to work throughout cancer, thousands of clinical trials. But the question that we had a while ago when I started in the program is, okay, could there be actual utility in using this same cell therapy where you're taking a cell and killing bad cells? Could that be used beyond cancer? I'll likely repeat myself a number of times during this talk. I like to think about things from first principles. So the question is, for something to work for at least traditional CAR T cell therapy, cytotoxic CAR T cells, what's needed? And I would say there's at least 2 prerequisites. The first is you need to have a bad population of cells. They can be directly being pathologic. They could be directly the things that are harming the tissue. It can be an indirect effect. You just need a population of cells that if you get rid of that population of cells, it does good things. And then super importantly, I think this is one of the reasons why we've struggled so much in the solid cancer setting is you need to be able to target that population of cells specifically. And though these are like very simple premises, they've actually been exceptionally delimited. If you think about blood cancers and solid cancers, identifying a bad population of cells that you can just, cancer is a little bit easier because cancer, we want to kill every last cancer cell. In a lot of other diseases, you don't just want to kill all of a bad cell type. Even if you have a pathologic cell, killing it may not be the answer. And then identifying suitable antigens has been exceptionally complex. And so I've mentioned some of the difficulties of CAR T cells in solid tumors. There's a whole bunch of unique issues in cancer that make it a really, really complex disease. Maybe one of the points I would want you to take away from this slide is this, that because CAR T cells have actually worked in cancer is really promising for other diseases. I think it is one of the most difficult indications for a cell and gene therapy to actually be successful. The reason is in contrast to a lot of other diseases, you have this rapidly exponentially proliferating cell population, right? It's under a huge amount of genomic stress, meaning that these questions of like antigen escape, losing the target CD19 or BCMA, these are real questions, and the cancer is actually under genomic evolutionary pressure to lose that antigen. Especially in solid tumors, you have this complex tumor microenvironment. You have this really, really high target burden. The first patients had pounds of tumors when they were given CAR T cells. And so for all of these reasons, targeting cancer is actually really, really complex. And the fact that CAR T cells can do it and can be successful is really promising. And one of the things that I think is interesting, this is again we were thinking about this more from a science or a discovery platform perspective, is in non-cancerous diseases, a lot of these hurdles, the reasons CAR T cells weren't working in solid cancers aren't actually there. So, in cancer, you need to get rid of every single last cancerous cell or 99% of every last cancer cell, or the patient is not cured. That's not true for a lot of other diseases. A lot of other diseases, if you can eliminate, 30% of the bad population, you're doing good things for the patient. You don't have this like weird architecture in the cancerous tumor microenvironment or immunosuppressive. In fact, a lot of other diseases, the cell types are endogenously recruiting the immune system. So these are all reasons why targeting non-cancerous cells, at least from a first principles perspective, could be advantageous. And so what I want to talk to you about, I'm not going to talk to you about all of these diseases. What I want to talk to you is the vast majority of academics and industry and pharma are focused on the use of CAR T cells for cancer. There's a huge amount of focus in blood cancers because of the clinical proof of concept. And then there's been an enormous amount of utility or discovery in solid cancers. And what I want to say is that is just the beginning. And one of the reasons, maybe I'll revisit this because I'm not gonna make the point on any slide, is one of the reasons why I think it's exciting that we're past an inflection point is because I think if you had tried to enter into autoimmunity or fibrosis or senescence, your rate of failure would have been so high that probably you couldn't actually explore these therapies. But now the field understands that these can be curative. These are extremely powerful and potent. And so I think it's exciting. It's an exciting time to know that if there's anyone interested in studying any one of these aspects or even commercially exploring them, this is something that is powerful and the field has an appetite for. So CAR T cells in autoimmune disease. When we first started thinking about this, we were thinking about it again from a basic science standpoint. Could we do some cool preclinical proof of concept that you could use CAR T cells beyond cancer? Honestly, we still thought we were probably 5 years to maybe a decade away from actually showing clinical proof of concept in a non-cancerous disease. And so a lot of most of the data I'm going to show you today is data from other groups that have published in the interim. So one of the reasons why autoimmune disease is the most mature of these fields is the exact same CAR T cell that we use in leukemia and lymphoma could be used to target B cells in autoimmune diseases that are driven by B cells. So in a number of patients with systemic refractory autoimmune disease, B cells are thought to be a main contributor of pathology. This actually could be a way that the disease is being, is actually driving the disease. And so the question is, could you use the existing CD19 or BCMA CAR T cells to target these B cells? So at this stage, there's well over 40 patients that have been reported across the literature. This is in a variety of different diseases. What I'm going to do is I'm going to focus on the first case series to just talk about the results that we're seeing and try to point out generalizations that can be made from this. So just for context, maybe I'll come back to this. For context, there was a single patient reported in the New England Journal in 2021 that kind of took the field by storm. A single patient who was given CD19 CAR T cells. She had lupus, and they saw really dramatic effects. And so that was interesting and intriguing. It caught everyone's attention. I think it was a surprise to many in the field that this was being clinically explored in autoimmune disease. This is by Georg Schett's group out of Erlangen. And the first case series was published in Nature Med. And I would say this is the point in time when the field really realized there's something here that's going on. So I want to talk about the use of CD19 CAR T cells in 5 different patients. Essentially what they looked at is do these CAR T cells actually expand in these patients? So maybe a number of interesting points to think about is, in contrast to cancer, an autoimmune disease, when you are actually taking out the patient's cells, expanding them in the lab, one of the questions you have to be at least a little bit wary of is, will the T cells actually expand? Obviously, patients who have had cancer treatments have gone through chemotherapies and maybe their immune compartment isn't as good. But if you have a patient with autoimmune disease, you have to at least be worried potentially, are you activating and expanding immune cells that could be toxic to that patient? They showed for all 5 of the patients that they treated that they were able to expand these cells, and that when they infused them into the patient, this is post-lymphodepletion, so all these patients were lymphodepleted, that you do see expansion of the CAR T cells. They peak at day 10, And in most of the patients, they go away within a month. This coincided with the rapid depletion of B cells from the periphery. So when they looked at the B cells in all of these patients, they saw a really rapid reduction of all B cells. So saying that the CAR T cells are not only expanding in the patients, but they're actually also functional in the patients. And this happened all the way out to a month. Now, what was remarkable is in all of these patients, they saw a marked and strong, essentially, amelioration of the disease. So whether you looked at clinical values, clinical indexes, so looking at double-stranded DNA antibodies, those disappear more or less in the blood by 3 months. Symptoms like fatigue are essentially down in every single patient. And if you look at, essentially the clinical indexes for these disease, in all 5 patients, by 3 months, they were declared to be in remission. One point I should make that is super important to make is these are patients who post-infusion of CAR T cells were taken off of immunosuppressive drugs. So for the vast majority of these patients, from the time of diagnosis, they have been on immunosuppressive drugs. They were given CAR T cells, taken off immunosuppressive drugs. In the absence of immunosuppressive drugs, by 3 months, they are in remission. Now, that's incredible, but one of the big questions that the field had is this, is what's actually going to happen to these patients long term? What's going to happen to the B cells? So for a good subset of cancer patients that are treated at these sites, they have chronic B cell aplasia, meaning that their B cells are gone long term. As you can imagine, if you have cancer, maybe that's an acceptable toxicity, but a significant toxicity. Patients walking around without B cells are much more susceptible to infections. And so these patients, I didn't do a great job of contextualizing. These are extremely sick patients with autoimmune disease, lupus. So these patients have life-threatening autoimmune disease. And so essentially, the investigators were just of the opinion, and rightly so, that if this comes at the expense of maybe chronic B-cell aplasia, that's a reasonable trade-off. Really shockingly, what this plot is showing you is the CAR T cells in the dashed line start to go away more or less by day 100, day 50. What is interesting is post about day 30, in a couple of months, the B cells start coming back in these patients. And so, the worry is, okay, if the B cells are coming back, the CAR T cells aren't persisting, or if they're persisting, they're not functional, do the symptoms come back? And what they saw is even though these B cells do come back, there is no, this data has now been taken further than 15 months for this one patient. There's no return of symptoms. So despite the fact that the CAR T cells are infused, the B cells get depleted for a few months and then come back, they get repopulated, there's no return of clinical symptoms. The next question that's super important is all of these toxicities that we really think about every day, what we're worried about when we're giving CAR T cells to patients, what happens to those? Do they get CRS like Emily got or patients get? And kind of what we had put forward that potentially, because there's such a lower target burden, you don't have pounds of B cells that you're eliminating like in leukemia or lymphoma, that potentially you would have less cytokine release syndrome. And that's what has now been seen across many patients is that there's not significant or severe cytokine release syndrome in most patients. There's not this huge spike of cytokines. So very mild and manageable CRS in these 5 patients. This was done, this is a follow-up study by the same group looking at 15 patients across 3 different diseases. Again, just to give you a context of how sick these patients are, this is a patient at baseline. The same patient post a single infusion of CAR T cells, taken off immunosuppressive drugs, and essentially have a complete resolution of the disease. So this has now been done by more than the group at Erlangen. There's a number of groups who have reported the utility of CD19 and BCMA CAR T cells across a wide spectrum of diseases. These are still case series. At times, it's just a single patient being reported. So phase 1 and phase 2, multi-center trials are needed to really understand what is actually going on in these diseases. But at this stage, we're talking about well over 40 patients who have been actually publicly reported across a wide spectrum of refractory autoimmune diseases. So it's clear that CAR T cells in a non-cancerous context have significant utility. So a really important question that we have is, will these therapies actually be able to reach these patients? Something we talk about or think about all the time is, if tomorrow there was a solid cancer indication that we could actually treat, how would you actually get all of these therapies to these patients? Because we just can't, we don't have the infrastructure to manufacture these therapies for all of the patients who would need it. That is especially true if autoimmune disease it proves to be as significant as it is. Like if tomorrow they said for severe forms of lupus, the FDA is giving approval to actually treat these diseases, we could not treat the patients who needed it. So there was a very intriguing report that was published last year of using allogeneic or donor CAR T cells for autoimmune disease, just in 3 patients. But I'm going to talk about a little of this. Most of you are aware of this, but it's essentially the idea of, instead of doing this autologous, using a patient's own T cells, engineering those, could we take a healthy donor, genetically modify them so that that donor could be infused into a whole batch of patients, and then essentially kept on the shelf? That would bring costs down, that would improve scalability, that would improve product-to-product homogeneity. And so the question is, could you actually use donor CAR T cells in autoimmune disease patients? And again, this was a group out of China. What they did is they showed that, again, in all of these patients, you see expansion of these CAR T cells, obviously to differing degrees, but we see expansion of CAR T cells across all 3 patients when you use this allogeneic strategy, CRISPRing out so that you don't have graft versus host disease or rejection. Again, if you look at the B cells, you see that the B cells, they disappear. And then in all 3 patients, by month 6, you start to see reemergence of B cells. And again, the question is, do these patients actually get better? And what you can see, whether it's in scleroderma or inflammatory myositis, these patients all have a complete response. And again, very similarly to what was seen in the autologous trials or the autologous reports, we don't see, these groups did not see significant toxicity, cytokine release syndrome or cytokine. So I don't know if I've done as good of a job as I wanted to in just like implicating how paradigm shifting this is. You're talking about, for some of these patients, for some of the patients that have been reported, they've been on drugs and having disease for decades. Single infusion of CAR T cells. Again, are these patients cured? We don't know. The first patient that was treated in 2021, as far as we know, I just talked to Georg not too long ago when he visited Penn, that patient is still in remission. So are a subset of these patients cured? The next question is, what's this reason for acute B-cell aplasia? We wouldn't have predicted this necessarily. We would have thought that there would be chronic B-cell aplasia. So we've spoken that at least one of the ideas that needs to be tested and explored is potentially the reason why when we look 10 years later in a subset of patients, we can see CAR T cells floating around in leukemia and lymphoma patients, is because the B cells make it to the bone marrow in leukemia and lymphoma, meaning the CAR T cells need to make it into the bone marrow in leukemia and lymphoma. And we think that maybe getting into the bone marrow causes these CAR T cells to become CAR memory T cells. But we wonder whether if in lupus or autoimmune disease that you don't need that expansion, those CAR T cells never make it to the bone marrow, and so potentially they don't persist. But what is this reason for acute B-cell aplasia? It is remarkable, and it is good for patients that this is indeed the case. Obviously, a very important question. I've told you only positive data because as far as I know, right now there is only positive data, but almost certainly there will be long-term consequences for some of these patients. So what are those long-term consequences? Are there safety risks? It's not the purpose of my talk today, but I'm sure most of you have heard about the question the FDA raised last year or 2 years ago now about the question of CAR T cells actually turning into cancer. Is that a real concern? Does that change the math of whether or not patients who have systemic autoimmune disease, should they get these therapies? Another question that we really need to wrestle with is for cancer, lymphodepletion prior to CAR T cell infusion is super important. Is that similarly important for patients who have refractory autoimmune disease? When you need such a smaller level of expansion of the CAR T cells, could that come just increase the bolus that you're actually infusing rather than lymphodepleting these patients? Those are real important questions that we need to answer. And then what is the true rates of response? Right now, we are aware more or less that almost all of the patients that have been treated have responded. There's only one that I know of who has not responded. They were infused with a different CAR T-cell product and have responded. And so what are the true response rates across multi-centered clinical trials? And those studies are being done now. Okay, so I'm going to pivot to talk about now not the use of CAR T-cells in autoimmune disease, but more exploratory preclinical use of CAR T-cells in fibrosis. So this work was done just before I started at Penn, out of the John Epstein's lab, who's now the dean at Penn. So cardiac fibrosis is a known contributor to heart failure. So as you all know, heart disease is one of the most implicated diseases in our population. And what you can imagine is a organ whose primary job to actually pump, having fibrotic tissue deposited in that, is extremely pathologic and debilitating. And so the question is after a heart attack or after some heart insult, your body puts down this fibrotic tissue to essentially protect your heart from rupturing, right? As you have cardiomyocyte death, if there is not fibrotic deposition, your heart could rupture. And so you need that in the short term, but the question is, in the long term, is that actually pathologic? And so John and his group had this idea of, well, is there a target on these activated fibroblasts and, in the fibrosis in the human heart that you could actually go in and target with CAR T cells? What you are seeing is a non-failing heart from a human on the left. And then as you go over two different types of cardiomyopathy, you can see FAP in green is expressed in these hearts. And so they did this really, really elegant and impressive study. It's actually what got me into the field. I thought it was really amazing. What they did is they gave mice heart disease through hypertension, angiotensin, phenylephrine, and then they gave them these FAP CAR T cells and asked, what happens over time to these mice? So what you can see here is a healthy heart on the left. And then when you give these mice heart failure, you have this scarring, this fibrosis, and that's depicted here in red, pico-serious red. And then the question is, if you give these mice FAP CAR T cells, what happens? What you can see is, at least in this mouse model of cardiac fibrosis, you see almost a complete resolution of the fibrosis. And this is accompanied not only by a histopathological reduction of the fibrosis, but also a rescue of heart function. Shortly after John's group, in collaboration with Drew Weissman's group, asked the question of, what if you did the same thing, except instead of using ex vivo, so taking the T cells out, making them in a laboratory, then infusing them back into the mice, what if you did this all in vivo? Could you essentially use the exact same technology that was used in the vaccines that were deployed and target the mRNA that encodes the CAR to the T cell, just by simply sticking an antibody in the lipid nanoparticle, so that instead of being taken up primarily by the liver, it would be taken up by the T cell. That mRNA then gets translated into the CAR protein. That CAR protein goes to the surface, and then you have a CAR that was made in vivo. You didn't have to do any of that complicated process. And obviously, again, similar to allogeneic, this drastically could improve cost and scalability, accessibility. And the one point I would just make for the targeted lipid nanoparticles is you also have a transient CAR. So that CAR disappears as soon as the mRNA disappears. It's not there forever. And essentially, just to summarize this data, they showed the same thing. You could have the exact same effect. So you have a healthy heart, you have a diseased heart, and just a single injection of the LNPs generated the CAR T cells in vivo and showed anti-fibrotic activity. The last thing I'll talk about, the last area I'll talk about, is senescence. So again, you have a pathologic cell type, cellular senescence. They play an important role in a number of diseases, as well as normal development. But essentially across the last decade, they've been shown to play a causative role in a number of different diseases, from lung disease to normal aging to metabolic disease. And so a group at MSK asked the question of, could you actually generate a senolytic CAR T cell to target drug or diet-induced liver disease? And so they identified this candidate antigen UPAR. They built a CAR against it. This is untransduced T cells. So again, the serious red is showing you fibrosis in the liver. SA beta gal is a marker of senescence. When they give UPAR CAR T cells to these mice, you can see a resolution of the fibrosis accompanied by a strong elimination of this marker for senescence. And this is accompanied by improvements in liver function or liver readouts. Okay, so one last thing I just want to mention is there is a lesson here from CD19 is probably the best target, at least given our current context, is something that you could use in cancer and show efficacy in cancer and then apply it beyond cancer. So CD19, one of the reasons it was had such an enormous impact is because it could be used in leukemia, it could be used in lymphoma, and especially those early trials from Erlangen used the exact same CAR, this exact same binder, the exact same co-stimulatory domains for autoimmune disease. And you can learn about the safety profiles, and you can learn about what's going on in the context of cancer patients and apply that to autoimmune disease patients. Okay, so to conclude, right now it's very well accepted that CAR T cells is a new pillar in oncology. Radiation, chemotherapy, surgery, targeted cellular therapies is a new pillar. The point that I want to make is I am entirely convinced that cell therapies is a new pillar of medicine at large. We're going to look back at these years and realize things have changed forever. And so I think what started in cancer will start this revolution. CAR T therapy, it is now, I think, clear, will expand beyond cancer. To what context and how far in autoimmune disease is unclear, but it's going to go beyond cancer. And one of the really exciting things is there's a lot of synergy here. The things that we'll learn in cancer and autoimmune disease will feed both. And so the ongoing innovation that's being done right now in cancer and in autoimmune disease will help each other. And so not just a new pillar of oncology, but a new pillar of medicine. With that, I'll thank both of my mentors in my labs, as well as many collaborators, both at Penn and abroad, and I'm happy to take questions.
Yes, we'll bring you a mic.
Thanks for the presentation. It seems to me that the comparisons CAR T cells with respect to deleting B cells in lupus to what has been done using monoclonal antibodies is a little murky because the monoclonal antibody was an anti-CD20, phase 1, 2, and 3. CD19 is very different as a target in that CD19 is expressed throughout the B cell life, development and peripheral differentiation, as well as plasma blasts and plasma cells, which are very relevant to the production of autoantibodies in lupus. So as you probably know better than I do, that for many different reasons, the CD20 in lupus has been abandoned because the results were not quite right, in the lack of a better terms. But I will argue that the major differences here is the target, 19 versus 20. I'm not sure it is the CAR T.
Yeah.
And I sorry, just to conclude, I always think about, I'm a B cell guy, and I always work. Before working on B cell, I was working with serum 50 years ago. And if you in vitro, human serum has 1,000 hemolytic units per ml compared to 10 in the mouse serum. So a monoclonal antibody can be quite effective in vivo, in humans.
Yes.
Need not be, I think in many cases, a CAR T.
I think it's a fair point, and I think we are too early on to say whether or not a monoclonal antibody or a BiTE or something like that couldn't be as potent as CAR T cells. So I'm telling you my impression and not what's true. I mean, like we'll need the trials to actually figure this out. But they've tried to do the head-to-head studies now of asking what is happening to the peripheral B cells. So not only the B cells in the blood, but also the B cells in the organs, post either bispecific antibodies or CAR T cells. What they're seeing is deeper depletion of the B cells in the peripheral organs as a result of the cell therapy, thinking that the infiltrating into those different organs is better. But we're talking about a small number of patients. This needs to be done with state-of-the-art B cells. So I agree that because the targets are different, those studies that actually need to be done using the appropriate bispecific modalities.
Hi, very inspirational talk. I'm not immunologist, I'm bioinformatician, but what you mentioned for me is very interesting when you said that one of the reasons why the solid tumors are not being treated or failed by CAR T cells because they are under evolutionary pressure to lose the antigen that makes them to be recognized. My question is, why do you think this doesn't happen in the cases that autoimmune disease that you have shown, or why you think it in the long term can happen and trigger, resistance like it happens with antibiotics or other drugs? Do you think that could be a potential scenario?
Yeah. So I think that antigen negative relapse could happen probably in any context. I think that in cancer, I think CD19, because it's a lineage antibody or a lineage marker, it's much less likely to be lost. But we know that it can be lost in cancer. In solid tumor settings, a number of the targets that we're looking at are not lineage markers. In fact, we don't have a great lineage marker. And so there's a lot less pressure, biological pressure, to keep that antigen. So if a cancer cell loses an antigen and stops getting killed, it will do that preferably. My sense is that won't happen with the same rapidity as it does in autoimmune disease, but time will tell.
I'm assuming this is on. Kind of a follow-up to what this gentleman was talking about. My impression of what you're saying, and I used to teach the autoimmunity part for graduate school, but you know far more than I do, so I'm going to give you that right off the top, is by coming in with CAR Ts against CD19, you're knocking down the entire B cell population. So is the patient and then going to end up having to possibly get these CAR Ts back again periodically? In a way, that's kind of what I figure is going on. So I'm wondering if the next step would be, you've got a B cell that's a pathologic B cell. It's making, say, the antibody against the desmin protein that's holding the two skin cells together. It's got surface IG. Why not target the epitope of that antibody and knock down the B cell specifically?
Yeah, that's a great suggestion and idea. And the answer is that about 5 years ago, people actually deployed this. This is called the chimeric autoantigen receptor T cell. So there's two reasons why that's complex. Very often, we don't know which B cells are actually promoting the disease.
Yeah.
Right. So in the context that you described,
and if you grab some, you're going to have probably leakage on the side.
Exactly.
But
And
anything from knocking down the whole B cell population.
exactly. But the one other point I would make is what is happening here, at least as what we're seeing, is there's this reaccumulation of B cells. But what the New England Journal paper from just recently showed is that is not accompanied by relapse of disease. You would think that it would,
Yeah.
but what the investigators are putting forward is this idea that you essentially maybe have an immune reset.
Yeah.
Whatever went wrong initially to give these patients autoimmune disease has now essentially been reset. You get the reemergence of these B cells, but these B cells are maybe not.
Your data didn't indicate a possible relapse, but then you said, you've got to wait a little bit longer for some long-term.
Exactly.
studies.
And some of these patients,
Very promising.
yeah, some of these patients may end up relapsing. And the question of could you just deplete the pathologic B cells, I think from a first principles standpoint definitely makes sense. But I think because the acute B cell aplasia seems to be the case for a lot of these patients, maybe the cost of doing that becomes more complex.
So our next speaker has a question. Just in the interest of time, I'm going to move on. Dr. Lionel, if you want to come up and finish your talk. So Dr. Lionel was, I guess I don't need this. Was recommended to me by Dr. Sa Wong. So he's here to present some work that they presented at another meeting. But I do have an announcement before he goes. Right at the end of this, we have our yearly picture. And so we'd like you guys to meet us out front. Is that right, Leroy? Okay. So just when this ends, before you go get your lunch, if you want to be in the picture, join us there. So thank you very much, Dr. Lionel, for speaking. And we're looking forward to hearing more about your talk. If you want to ask your question, you could.
Yeah, I think I can wait. Thank you for the opportunity to share our work on some of the clinical applications of flow cytometry based quantification of CAR T cells in the clinical setting as an early prognostic biomarker in lymphoma. So this is work in Dr. Anil Poo's research group at the Department of Lymphoma at MD Anderson. So within the broad umbrella of lymphoid cancers, we have Hodgkin and non-Hodgkin's lymphoma, and then within non-Hodgkin's lymphoma, we have B and T cell lymphomas. And then among B cell non-Hodgkin's lymphomas, diffuse large B cell lymphoma is the most common aggressive type of non-Hodgkin's B cell lymphoma. So it represents just under half of all non-Hodgkin's lymphoma globally. And in any given year in the US, there's around 30,000 newly diagnosed DLBCL patients. The incidence does vary among different ethnicities. For example, it's lower in East Asia compared to Europe and North America. So the good news for DLBCL is that about 2/3 of patients with DLBCL can achieve a long-term remission or essentially like a functional cure after first-line therapy, which is usually with chemotherapy. And that's been the case for roughly 2 decades since the anti-CD20 antibody called rituximab was introduced in the clinical setting together with less targeted chemotherapy. However, for the 1/3 of patients who relapse with lymphoma after receiving the first-line treatment, the options have typically been more limited. There are also around 10% of patients who have refractory disease, so they never go into remission despite getting the first-line chemotherapy. So in patients who have either relapsed or refractory DLBCL, the outcomes have tended to be relatively poor. If left untreated, relapse or refractory DLBCL has a life expectancy of around 3 to 4 months. So this has been a clinical space that has been the focus of a lot of research, and as very elegantly outlined in the previous talk, there's been a lot of progress in immunotherapy and CAR T cell in this space, which is one of the fastest moving in hematological malignancies. So as everyone in the audience is very familiar with the CAR T cells, you have the T cell extraction from the blood of patients. This is autologous CAR T cell manufacturing. And then there's usually a period of 3 to 4 weeks where it's sent to the commercial CAR T facility where you have the genetic engineering to insert the chimeric antigen receptor in the T cell. And then usually about after a month from when the patients had the blood extracted, they get CAR T cells back. And then in lymphoma, the CAR T cells have the target of CD19. So the CAR T cells go to work and they expand, and with the goal of essentially wiping out all lymphoma cells in the patient. So starting about 8 years ago, there has been enormous progress in getting CAR T cells to thousands of patients with B-cell lymphomas. So starting initially with Tisagen Lechlucil, the brand name Kymriah, initially in the pediatric B acute lymphoblastic leukemia space with that index patient that was presented in the previous talk, followed shortly thereafter by Axicel or Yescarta in adult relapse refractory large B cells. So that was 8 years ago. And since then, there have been several products which have been approved by the FDA for both large B-cell lymphoma, such as Yescarta, Kymriah, and Breyanzi. And there are also products for mantle cell lymphoma, for multiple myeloma, and again, for B-cell acute lymphoblastic leukemia. So another recent development has been that initially these products were in the 3rd or later line setting. So a patient had to have a disease that was progressing despite having 2 lines of treatment. Lately, the CAR T cells are moved further up in the treatment pathway. So if you fail even 1 line of therapy for diffuse large B-cell lymphoma, you can now access CAR T cell, especially if the relapse happened within a year after the initial chemotherapy. So the algorithm for the treatment of large B-cell lymphoma has been evolving rapidly over the past 2-3 years because of pivotal trials that have shown that CAR T cells actually have a survival benefit compared to the pre-existing standard of care, which used to be autologous stem cell transplant. So now, if you had first-line chemotherapy in large B-cell lymphoma and there was a relapse in less than a year, which 75% of relapses tend to happen within a year of first-line chemotherapy, then they would be eligible for CAR T cell that would be covered by most insurances in the US. And then out of that, 70% of patients would typically get CAR T cell therapy. There are two agents which are widely used now because they have solid survival benefit over autologous stem cell transplant. And then in about 30 to 40% of them, you have a 5-year or greater long-term remission, which is the functional equivalent of a cure. There is a fraction of patients who aren't eligible for CAR T cell therapy, mainly because of other comorbidities or performance status. So it's now, CAR T cell is FDA-approved standard of care in patients with relapsed diffuse large B-cell lymphoma. But unfortunately, more than 50% of patients who receive CAR T still have relapse of the lymphoma after CAR T. In the clinical setting, you kind of assess if the lymphoma has responded to the CAR T cell therapy by doing PET CT scans, typically 3 months after the date of the CAR T cell infusion. So you usually wait 90 days and then you do a PET CT scan and you compare that with the scan prior to the CAR T and to see if the lymphoma is gone or if it's still present or progressing. And in addition, as mentioned in the previous talk, there's also severe toxicities associated with CAR T cell, such as cytokine release syndrome, as well as a fairly specific neurological toxicity called ICANS, which is caused by the immune effector cells crossing into the CNS and causing this constellation of symptoms that are grouped under the umbrella term of ICANS. So CRS, as was mentioned in the previous talk, is because of release of several different inflammatory proteins such as IL-6 and interferons, which can present with fever, hypotension, need for oxygen support, and in severe cases, they end up in the ICU. And thankfully, this is less frequent these days, but can also, in rare cases, lead to death. ICANS is a bit of a harder toxicity to treat because it starts with a constellation of neurological symptoms, including encephalopathy, cognitive deficits, seizures, and one of the most severe consequences can be brain edema and herniation, which can be fatal. So there was interest in trying to see if we can use CAR T cell quantification as an early prognostic biomarker to predict response, as well as to see if we can kind of predict which patients might be at high risk for developing severe toxicities. So rather than waiting 90 days after the initial CAR T cell infusion, can we get an earlier preview of whether a patient might respond or not in terms of their lymphoma? And also, can we kind of like highlight those who might be at especially high risk for some of these serious side effects? So usually, there's a fairly predictable expansion curve for CAR T cells once they're infused into the patient. So on day 0, you get the infusion, and then they build up in the blood. Usually, the peak is between the end of the first week after the infusion to the end of the second week, so somewhere between day 7 to day 12. And then the CAR T cells go back down in the blood, and they persist at a very low level in the weeks after infusion. And just to provide context for the timeline of the typical toxicities, CRS tends to be the cytokine release syndrome tends to start within the first few days after infusion. ICANS tends to start a bit later, and a more rare side effect called the hemophagocytic lymphohistiocytosis, or extremely high inflammatory, extremely high hyperinflammation syndrome. It's a bit later than the other two side effects. And then, as was mentioned in the previous talk, there's B-cell aplasia and prolonged cytopenias, as well as decreased production of immunoglobulins that can last for months and sometimes years after CAR T cell therapy, leading to increased risk of infections. But essentially, there's a lot of interest into seeing whether we can use quantification of CAR T cells in this, the first week or two after the infusion, and kind of use that as a proxy for response instead of waiting till day 90 for the PET scan. So this has been studied in the past a few 2 to 3 years across different settings. So this from a table, from a review that we wrote on the topic. So there were initially clinical trials that were responsible for CAR T cells being approved in different lymphomas. So typically, these trials used qPCR, quantitative PCR, from blood samples of patients to actually obtain CAR T cell quantified levels, and then see if that correlated with things like neurotoxicity, with cytokine release syndrome, and also most importantly with response to treatment. So the one area on which pretty much every study out there has consistently agrees on is that if you have increased CAR T cell expansion, it seems to be associated with increased risk for this ICANS neurotoxicity. But when it comes to response to treatment, there is actually conflicting evidence from different studies. So many of these initial trials in mantle cell lymphoma, in follicular lymphoma, and large B-cell lymphoma, they found that increased CAR T expansion is also associated with like a better response to therapy at the day 90 PET scan. But other studies, including outside of the clinical trial in the standard of care space, in large B-cell lymphoma, mantle cell lymphoma, have not found a relation between increased CAR T expansion as well as response to treatment. So this still remains sort of like a question that needs to be addressed. And in addition, what are the factors pre-existing in the patient before they get CAR T cells that could affect how well they expand? Those are also not well understood. So this is kind of like led to our study where we were trying to investigate the prognostic utility of in vivo flow cytometry-based CAR T cell quantification for, again, to see if it definitively associates with predicting response early, as well as for risk for complications. So for this, in order to kind of address this question, we wanted to avoid some of the pitfalls in the previous studies because they had sometimes used multiple CAR T cell products or span different disease subtypes or lymphoma subtypes, or there were also like inter-assay differences between studies. So we wanted to use a single CAR T cell product or a single lymphoma subtype using a validated flow cytometry assay at precise time points. So the flow cytometry assay is an 8-color flow assay, which essentially like the key element is an antibody against FMC63. So these are the commercially FDA-approved CAR T cell therapies against the CD19 target in large B-cell lymphoma. So the FMC63 is a part of the extracellular ligand binding domain, or the SCFV, of all commercially available anti-CD19 products. So it's actually of mouse origin, so it's not found in physiological human T cells. So this assay was validated by Dr. Sa Wang and the hematopathology team at MD Anderson Pathology Lab for clinical use with the BD FACS Lyric flow cytometry system. So just in one slide to summarize the validation, it was done in terms of for accuracy using cell line testing with both the positive and negative cell lines. So the positive cell lines had both the CAR as well as the NF80 luciferase reporter, whereas the negative cell lines only had the NF80 luciferase reporter. So as you can see here in the positive cell line, when the FMC63 was part of the flow panel, and just looking at the CD3 positive T cells, you have a population that lights up. Whereas this is when we use the positive cell lines without the FMC63 as part of the panel, that is absent, and also the negative cell line does not have that population. So the other way of looking at accuracy was to actually use blood sample testing from patients. So you had patients who had received CAR T cell therapy, and in all 20 samples tested from such patients, the assay was able to detect the FMC63 positive T cells. And when we looked at patients who did not receive CAR T therapy, there was no detection of these FMC63 positive cells because, again, this is of mouse origin and not found in physiological human T cells. So in terms of precision, we also looked at running this on different instruments and at different times of the day and using different operators. So this has now been part of standard of care CAR T cell therapy for around the past year and a half at MD Anderson. So on our lymphoma CAR T ward, we were able to get data from 100 patients who received standard of care axicabtagene ciloleucel, which is also known as the Yescarta, which is the most widely used CAR T cell therapy for relapsed DLBCL at MD Anderson. So again, day 0 is the CAR T-cell infusion. And then starting on, so patients are typically admitted for a minimum of 7 days and a median of 12 days after CAR T-cell infusion to monitor for those side effects. So starting on day +1 and on every other day, we collect a blood sample that is sent to the lab for the flow cytometry. And the turnaround time is usually about like a couple of days after the submission of the sample. So as long as they're an inpatient, they get blood samples sent every other day. And then once they are sent home, they come back for clinic follow-ups. So they get outpatient testing on day +21 as well as day +28. And again, our response endpoint is the standard of care PET CT scan at day +90 after the infusion. So the median number of days with flow cytometry data per patient was 9, with a range of between 7 to 10 days with flow cytometry data. So we presented this at the ASH conference a couple of months ago. So this slide essentially shows the quantified CAR T cell levels. So on the x-axis is the days after the CAR T cell infusion, starting on day 0, and on the y-axis is actually the level of the CAR T cell in the peripheral blood in cells per microliter. As you can see, the cells don't really like show up on the day 1 or day 3. So these are like median data points. So then starting on day 5, you start to see this like cells start to emerge, and then you start to see this the peak that usually happens within sometime between the 1st to 2nd week after infusion, and then the numbers decline. So the two curves here, the blue is the responders. So these are patients who on the day +90 PET scan didn't have any signs of lymphoma. So they had a complete response to the treatment. Whereas in the orange here are the non-responders. So they either had stable disease, which didn't change much with treatment, or they had progressive disease, which got worse compared to before treatment. So we had 81 patients for whom we were able to have day +90 PET CT scans, and 58 responders and 23 non-responders. And as you can see, there is a separation quite early, starting from day +5. Then it kind of widens at day +7 and so on day +9. So these are significant differences in the median between the responders and the non-responders. And in addition, the peak for the responders also happened at an earlier day and at a higher peak CAR T cell value compared to the non-responders. So in terms of our statistical analysis, we were trying to determine which day after infusion had the best predictive value for response. So this is the logistic regression analysis that we looked at for day +5, 7, and 9. And the day +7 CAR T level had the highest area under the curve for the logistic regression analysis in terms of being associated with response at day +90 after CAR T cell therapy. And specifically, CAR T cell level of at least 30 cells per microliter had the best cutoff value to help predict responders versus non-responders. So the sensitivity was 78% and a specificity of 75%. And a different way of looking at it is just to split the responders and the non-responders. And the orange is a high expander, so greater than 30 cells per microliter. So you can see that the majority of the responders had more than 30 cells per microliter at day +7. Whereas the green is the lower expander, so less than 30 cells per microliter, day +7. So the majority of the responders like exceeded that threshold of 30, whereas the majority of the non-responders were below that threshold of 30. So at day +7, you kind of get a snapshot of how this patient might potentially like respond on that day +90 PET scan. So to also control for potential confounding factors, we did a multivariate analysis looking at other factors that are known from previous clinical experience and literature to be associated with response to therapy. So factors such as the stage or how advanced the lymphoma was on the PET scan prior to the CAR T, whether the lymphoma had continued to progress or was stable prior to the CAR T. The IPI is a clinical score that is assigned to patients with lymphoma to determine if they're high risk versus low risk. So it includes things like age and the LDH blood value, as well as performance status and like how widespread the lymphoma is. And finally, the LDH is a widely used proxy marker for disease burden and tumor volume in lymphoma. So we could see that even in the multivariate analysis, the D+7 CAR T level continued to be like highly significantly associated with response, even after controlling for the other covariates with a pretty high odds ratio for responders versus non-responders. And we also looked to see if there were pre-existing patient characteristics that were associated with having a higher D+7 CAR T cell level in the blood. So we did several univariate analysis across different potential factors that might play a role in having a higher D+7 CAR T cell level. So the ones that were significant initially in the univariate included like more advanced stage lymphoma, whether they received what's known as bridging therapy or therapy right before they got the CAR T just to keep the disease under control while the patient was waiting for the CAR T cells to come back from the commercial facility to get infused. And there's recent research that shows that exposure to the chemotherapy agent bendamustine actually negatively affects CAR T cell function. So we wanted to see if exposure to bendamustine was also affecting the D+7 CAR T cell level in the blood. The one factor that was actually significant in this multivariate analysis was whether someone received CAR T early on in their disease course compared to later on. So for example, second line patients would be those who received only one type of chemotherapy, and then CAR T was the second kind of treatment they got for the lymphoma, whereas the later line CAR T would be like third or fourth or fifth line CAR T. So usually by that point, a heavily pretreated patient has like issues with the T cell fitness, and this can also affect the CAR T cell function. And you can see that second line CAR T actually had higher D+7 CAR T cell levels in the blood. So we also looked at the association between D+7 CAR T cell levels in the blood and different kind of complications. So one of the things that stood out was ICANS, which is the neurotoxicity. There are different grades of ICANS. Typically, someone with grade 3 or 4 ICANS would end up in the critical care unit or the ICU. So this is someone who often becomes non-verbal, non-responsive, can have seizures, and more rarely they can have like cerebral edema and life-threatening complications of like herniation of the brain. So we can see that in the higher the D+7 CAR T cell level, there was also a significantly higher incidence of these the grade 3 ICANS. So this is something that's been shown in several previous studies, as I showed in that table. There was also an association between a higher D+7 CAR T cell level requiring ICU care during the admission for CAR T cell infusion. And finally, tocilizumab, which was mentioned in the previous talk as an anti-IL-6 agent that's used for treatment of cytokine release syndrome, was also like there was a higher use of tocilizumab associated with a higher D+7 CAR T cell level. To sort of summarize, higher D+7 level is a bit of a two-edged sword. You have a higher response with a higher D+7 CAR T cell level, but you also have a higher incidence of some of these CAR T specific complications with a higher D+7 level. So to summarize, we use like a validated assay, a single type of CAR T and a single lymphoma subtype to provide evidence for prognostic utility of blood CAR T levels as an early biomarker that even as early as D+7 after infusion can help predict which patients might respond versus not respond to CAR T cell. So this has clinical implications because we have a protocol for how frequently these patients are to come to clinic as an outpatient leading up to that D+90 PET scan to see how they're doing. Whereas if we have their D+7 level or their quantified CAR T levels and they're predicted to be non-responders, you can actually like keep a closer eye on them, and they might need to have a different type of treatment started earlier than D+90 because they are at high risk of not responding to the CAR T at D+90, but you can kind of catch them earlier and potentially intervene faster than waiting for the PET scan and then making your decisions based on that. And then this also has a potential implication of being able to highlight those patients who have a higher risk of having these complications even while they are still an inpatient, if they have a really high D+7 value. So there are different measures that you could potentially actually start in these patients as prophylaxis against these complications. So with that, I would like to acknowledge several faculty in the lymphoma department at MD Anderson, many of whom submitted patients for this study, as well as especially Dr. Neil Booth, who's the primary PI on the study, and Dr. Sa Wang and Wei Wang in the hematopathology department, and our colleagues in biostatistics and cell therapy. Happy to take any questions.
Right? I remember when we were just talking about doing MRD work and shifting to basically give that information back to oncologists. I want to call out, I think a real benefit here is to the patient themselves and their family to be able to get some kind of indicator along the way. Do you know how easy is this going to be to pass on to other institutions? Have you guys tried to do that? What was the reception like at ASH?
Yeah, I think one of the advantages of flow cytometry is that it's a technique that's widely accessible across different centers. So now that the assay is validated, we are trying to get combined with other centers to kind of get this out there in a publication form, which can help to kind of have a greater impact in the clinical setting and have more uptake across different institutions.
And then make changes in therapy decisions based off of it. So I think it's kind of like MRD on, like the what we call MRD clinical flow. This is the next thing.
Yes.
Cars. Okay. Any questions? Yes.
So I apologize for my ignorance, but in diffuse large cell lymphoma, do you reach durable B-cell ablation with CAR-T?
Yes.
You
yeah.
do?
So at the 90-day mark, something like 70 to 80% of patients would typically have no signs of lymphoma. And at the 5-year mark after CAR-T, about 40% of patients who receive CAR-T don't have lymphoma.
And they do have B-cells.
So B-cell aplasia is unfortunately, a complication of CAR-T cells. And in many patients, it can take up to a year for the lymphocytes to recover. So they're often on antivirals and antibacterial prophylactic therapies until at least a year. There are a few patients in which there's prolonged B-cell aplasia, even lasting years after CAR-T, but those are thankfully in the minority. Most patients, it should come back by a year.
So when you see the B-cells back, has there been any analysis to see whether those B-cells do have mutations in TET2, the catalytic DNA demethylator, because that is associated and has been shown in mouse to underpin the diffuse large cell lymphoma transformation?
Yes, that's an excellent question. And I think it's something that's especially relevant right now because, as you know, the FDA has this black box warning for CAR T-cell therapy because of few cases of T-cell lymphomas that have arisen after CAR T-cell. And one of the common findings in these case reports, several of which have appeared in the New England Journal of Medicine, is that they have these TET2, DNMT3A, and the kind of these somatic chip mutations, which there seems to be a kind of a clonal cytopenia piece that is underlying some of this B-cell aplasia and potentially this development of this T-cell lymphoma as well.
Thank you.
All right, guys. So meet us out for a picture. It's lunchtime, and thank you for a great talk. Thanks for a great session.